Here we go. Welcome back, everyone. Here we are in the course on fundamental principles in Catholic bioethics, the Masters of Bioethics at St. Thomas University. Always beginning with a little prayer. In the name of the Father, the Son, and the Holy Spirit. O Lord, send forth your Spirit, and we shall be recreated. You shall renew the face of the earth. Amen. Father, Son, Holy Spirit. Okay, before going forward, any questions or comments from previous lectures or other stuff? Basically, what I have is the usual, which is to keep up with your hours of internship, because slowly but surely the weeks are going by, and before you know it, it will be December. And so it's what is it? It's a total of 150 hours, so they can accumulate. And therefore, please keep up with your hours and send me those Excel spreadsheets by Friday midnight, right? Friday midnight. That way I can log them. It's a reminder for all of us. Okay, anything else? All right, so we're going to go forward now. Yes, the last lecture, I had prison ministry, so I was not able to do it in person. I'd asked you to see a video. I hope you saw it, and the PowerPoints were there. But it was basically about origins. So I'm going to do a very, very brief summary of that lecture. First, I'm going to turn off some lights here for the best contrast. And origins, we talked about the three big origins, right? The origin of the universe, the origin of life, and then the origin of the human species. And you can see that each one of those has its own set of excitement and complications, and layers of complexity, but they have been figured out pretty much. And it's a combination between science and faith, really, because they complement each other and they give us the fuller picture. The origin of the universe, about 14 billion years ago. Remember, we did the exercise of counting to 1 million, just intellectually by the numbers, and then counting to 1 billion, which took several years, day and night, going straight through 24 hours a day. So 1 billion is a huge amount, right? Recently I saw in the news that the US budget is now going to be in the trillion of dollars, which is an American trillion, 10 to the 12. These numbers are so huge, as I mentioned before, that we don't even have an agreement worldwide on how much is it, because a billion for us in the US, 10 to the 9, is 1,000 million in Europe. And then 1 trillion for us is 1 billion for them, which would be 1 million, right? And that's another reason why we use scientific notation, because 10 to the 6, 10 to the 9, and 10 to the 12 is the same quantity no matter where we are on Earth, all right? And what language you speak and so forth. So mathematics, really, I'm reading a book now that's called Knowledge and Mathematics. The basic argument of this book is that it's all mathematics. It's all mathematics. Everything can be reduced to a formula. You know, material beings, energy, anything that we know and we have yet to know can be distilled down to mathematics, okay? And I make that point because, especially for the youth, math is one of the challenges and science is the other challenge as we grow. But really these two skills open up tremendous horizons and opportunities for us as we go into the professional world. So the original idea was, 14 billion years approximately, 13.7 billion years is the age of the universe, which means that there was an origin, and that origin, which popularly today is known as the Big Bang, was a theory produced by an astrophysicist. Anyone remember his name? Belgian guy, Georges Lemaitre. Georges Lemaitre, he was a Jesuit priest also. So there you go. He There's a little quote, he had many quotes, but one famous little quote of his was that some people study the universe from the scientific perspective, others study it from the religious perspective. I have chosen both, he would say, right? Because he was a scientist, he was an astrophysicist, and he was also a Catholic priest. Like Gregor Mendel, for example, the father of genetics, he was literally a father, he was an Augustinian priest. Anyway, so the issue of the origin of the universe, the Big Bang, in physical terms or in astrophysical terms is known as the moment of singularity, which is also the mathematical definition of a point, right? Not a period, like I can take a pen or a pencil or a marker and make a period, but there's actually mass on that period, right? If we go at the microscopic level, there are molecules there. However, a point is a one dimension, and we can only imagine that with the mind's eye. We can see two dimensions. Two dimensions is X and Y coordinates, right? A surface, a plane, as is known in plane, plane, it's two dimensions. And three dimension, obviously, volume, three dimension, X, Y, Z coordinates. So we can imagine that also. What's the fourth dimension, by the way? And this is not science fiction. This is not spirituality. There's a real fourth dimension that is so obvious. We're all embedded in it. It's so obvious that we take it for granted and we don't realize. But there's simultaneously a fourth dimension with three dimension, which is volume. The three dimensions is volume, right? X, Y, Z. That's volume. But so stuff has volume. How about the fourth dimension? Time. Time. We are necessarily embedded in time. We cannot get out of time, not on this side of paradise. And time is going on all the time, and it's only going forward. And so we're no longer at the same place where we were. Well, it depends. So it's a frame of reference. It's another one that's important. I'm just shooting out stuff to get. This is the stretching exercise before the race or whatever, right? It's the mental stretching exercise that we're doing, that I'm doing anyway, to get me into gear. Let's see. So yeah, time. Frame of reference is frame dependent. Any observation that we do is frame dependent. Locally, meaning within this classroom and even within the building and within the city of Miami, right? We are in the same place, the three of us, and the other two who are, well, virtually you're in a different place, the two of you. But let's say locally, here in this classroom, the three of us are in the same place, locally. But when we consider a larger perspective, like, for example, the rotation of the Earth, we're no longer really at the same place with regards to the rotation of the Earth as we were 10, 15 minutes ago when the class started, because the Earth has rotated already for 20 minutes, right? I'm looking at the clock. And in 24 hours, we'll make a full rotation and come back where we were in relation to, let's say, the sun rays. We use a sun ray, a ray of sun, as a vector, as a point of reference. We are rotating, and within 24 hours, we'll come back to that point. But we're displacing the local place with regards to a larger frame of reference. And then we can augment that because our solar system is also in motion. It's in one of the arms of the Milky Way, way out there in one of the arms of the Milky Way. And that's a galaxy that is also rotating. It has a spiral form, right? The typical form or shape of galaxies is a spiral. What does that tell us? That they're rotating. The intuition is that they're rotating slowly but surely, actually very fast, that the distances are so huge that we don't feel it. Like the Earth is rotating about 6,000 miles an hour, something like that. It's just insane. The fastest thing on Earth is Earth. It's Earth itself, the spin, right? The rotation. But even that 6,000 miles per hour is insignificant with regards to a translation. The translation is around the sun, which we do in 1 year, right? But that distance, it's again so huge that that's about 60,000 miles an hour or something like that. That the Earth spinning is moving around the sun. We don't feel that drag either, thanks be to God, because the atmosphere is held in place and we're moving. The atmosphere moves with us, so we don't fly out. It's stuff that I've been reading in that book, Knowledge in Mathematics. Anyway, so you see, it depends on the frame of reference. But there's constant motion and dynamic. I wanted to introduce that fourth dimension, which is actually an amalgam. And that was one of Einstein's insight also with his theory of relativity, especially general relativity, which is that time and place are together inextricably, all right? So he talks about the time-space continuum, time-space continuum. So back to the origin of the universe, two main steps. The first one, inflation, which took milliseconds, like 10 to the 20 minus 25 seconds, right? 10 to the minus. What's 10 to the minus 10 second? 10 to the 1 second is 1 second, right? 10 to the minus 10, an order of magnitude, is 1/10 of a second. What do we use 1/10 of a second? And we use that. Actually, there are chronometers that can measure 1/10 of a second. It's every 4 years. I'll give you a clue. And another clue, this event rotates throughout the Earth, different countries. The Olympics, they go down to 1/10 of a second, right? The races and all that, because everything is they're all running against time one way or another. It's like we're fixating to run against time. But now Olympics, they can be measured down to 1/10 of a second. And that's as far as they're going, because 1/100th of a second is insignificant, humanly speaking, as far as human motion is concerned. But 1/10, yes. Anyway, that's 10 to the, that's second to the minus 10, right? Then to the minus 20 to the minus 40. So that inflation that happened, that first event from the Big Bang, from 0 to 10 to the minus 23 or 24, I think it is, in milliseconds, right? That's the huge expansion, but it's called inflation.
So what's a nanosecond?
A nanosecond is 10 to the minus 9.
A nanosecond is 10 to the minus 9?
Yeah, 10 to the minus 9. Yeah. And then what happens is that inflation, during that inflation is where the quarks were formed. The quarks are the subunits of the subatomic particles. The three classical subatomic particles are what? They make up the atom. Hints. Yeah, proton, neutron, and electron, right? Two of them have charge, one doesn't have charge. All three have mass, but the electron has a negligible mass. It's not that it doesn't have mass, because if it doesn't have mass, it's pure energy. But it's not pure energy. It's 99.99999 energy, and it's got a little mass, but it's negligible. Anyway, by themselves, they're not stable. They're flying out all over the place. But when they come together, they coalesce and they form a stable unit, which is called the three subatomic particles coming together, forming a unit called the atom. And this atomic theory already was in the mind of the Greeks. Classical Greece, we get so much from classical Greece 400 or 500 years before the time of Christ. Of course, it was all theoretical. They didn't have proof of it, but they intuit that if they take a rock and chop it and break it up, they get little pebbles. And if they break those, they get sand, and you can still push the sand and get smaller and smaller particles. So they figured that there must be an elementary particle that they cannot see yet, but it's there, right? And that was their reasoning, the fundamental, so the basic unit of matter that is stable, the atom. To this day, 2,500 years later, paradigm still holds. Of course, Niels Bohr, contemporary of Albert Einstein, gave us what we call the standard model of the atom, which is that, all right? And it still holds today, the standard model. Anyway, what happened in those nanoseconds, that explosion of the Big Bang within fractions of a second, is the particles that make up the subatomic particles, the particles that make up protons, neutrons, and electrons. And now they've discovered also antimatter like positrons, which is an electron with a positive charge, basically, and so forth, right? All the particles that make those, and those are called quarks in general, quarks. They form at that time. And then, little by little, those quarks started coming together and forming the subatomic particles. And then the subatomic particles started coming together to form atoms and so forth. And we see with atoms already, we have the beginning, the weak force, the strong force, and as atoms coalesce together, they form molecules, and then the beginning of gravity eventually will come into play. All right, so the four fundamental forces, keep in mind, two of them are super tiny that basically stabilize the atom, strong and weak force, and they're also known as interactions. And then the two are the super macroscopic scale, the other two, which basically regulate the systems of the universe and stabilize that. And those are gravity and electromagnetism, which are also known as fields. Then the second part of that origin of the universe, after the few nanoseconds, nanoseconds, it goes into expansion. So inflation first and then expansion. And expansion is measured in the opposite scale of time, which is millions or billions of years. And that's how we get to the 14 billion. Right? So you notice that there's a very fast explosion, and then everything slows down to an almost still point, but not totally still moving, and actually accelerating. So it gets very quirky out there. It gets very esoteric, very crazy, can't find the right word, non-conventional. It's totally out of our league, out of that middle world that we live in, because it's beyond our senses totally. We need instrumentation, ever more sophisticated instrumentation to measure, just like measuring the microscopic world, the super microscopic world, all right? But expansion continues, and there's an acceleration of the galaxies away from each other. Another analogy that I saw at some point, hearing some tapes on Einstein, to picture it. For example, imagine raisin bread. Before we put it into the oven, we have the dough and the little raisins inside, right, all over the place. It's a three-dimensional image. So we put the raisin bread inside the oven. What's going to happen to that whole mass? It's going to expand. So we can see with our mind's eye that the raisins, all of the raisins inside the raisin bread, are getting away from each other proportionately as the bread rises, okay? But it's not at a constant speed, it's at an acceleration, and that's what's happening to the universe. And that brings a whole other set of questions and unknowns and stuff like that, that is really exciting, and for some people troubling, because we cannot really get a grasp on it. All right, that's the origin of the universe, and in there somewhere is this little tiny galaxy called the Milky Way, out of trillions of galaxies. And within that galaxy is one little solar system that we just call our solar system, for lack of a better name, in one of the arms of that Milky Way. And within that solar system of the 8 planets, the 4 super planets, the 4 smaller planets or inner planets, and then those other 4 microplanets we discovered so far, is planet Earth, which so far the only one we know of that has liquid water, and therefore the possibility of organic life as we know it, right? As we know it, which is within a liquid environment, within a water environment, and it's carbon-based, and it's made up of these 4 organic compounds. So you see where I'm going with the organic compounds, the Urey-Miller experiment, which pretty much gives us a theory for the origin of life. Again, life meaning organic life, not the life of the saints or the Blessed Trinity or anything like that, but real organic life as we know it here on Earth, which is made up of 4 main groups, or you want to call them domains, or, well, kingdoms, which would be the one that we belong to organically, biologically, move around, reproduce, well, all of them reproduce, but have locomotion, and we are consumers, we're not producers because we don't photosynthesize. Again, so obvious, we miss it, the animals. The other group, producers, photosynthesize, the plants. And then a couple more that are microscopic or somewhat microscopic, the fungi and the bacteria. So those are the four large kingdoms that I'm using as far as the simplified view or classification of life on Earth. All beginning with organic compounds, right? Those four organic compounds, proteins, lipids, carbohydrates, and nucleic acids. We should know this in our sleep. How to get to organic compounds from inorganic compounds. That's what the Urey-Miller experiment does. It gives us a possibility of generating with the primitive atmosphere, which is very different from ours, a lot of methane, a lot of ammonia, a lot of, well, some CO2, carbon dioxide from all the volcanic activity going on, water vapor. You mix those, it's a very toxic atmosphere. But the big important thing about the primitive atmosphere is that there was no free oxygen, no diatomic oxygen. What am I talking about? Chemical formula for diatomic oxygen, O2. We're breathing it right now. It's about 60% of our atmosphere. And it's keeping us alive. It's keeping all matter alive, all organisms alive because of cellular respiration that needs oxygen, right? O2 is in gas form at this temperature and pressure and so on and so forth. There was no O2 in the primitive atmosphere. But there was, again, methane, ammonia, carbon dioxide, and water vapor. Combination of those, some of which are organic compounds and some are not organic compounds. For example, water is not an organic compound because it's got hydrogen and oxygen, right? But it's missing what? To be an organic compound. Remember, organic compound, you need two elements, carbon and hydrogen. Don't forget about hydrogen. It's a minute amount in the atmosphere. It's a good thing because it's highly explosive, but it's there. And hydrogen, remember, hydrogen coats, basically covers most organic molecules and stabilizes them, right? Hydrogen is basically a proton. It's a positive charge to kind of balance all that negativity that is on the surface of many organic compounds. So water, H2O, there's no carbon to be seen. Conversely, CO2, there's no hydrogen to be seen, so CO2 is not an organic compound either. And yet they are vital, they're very much involved in life, right? H2O and CO2, but they're not organic compounds. They're precursors and they're needed for life to occur. All right, anyway, what Urey and Miller did is they mimicked the primitive atmosphere from what we could tell more or less of how it was, and they simulated a lightning. What was the lightning? Simulated in the jar, in the thing, the contraption that they built. It was very simple. A spark between two poles of electricity, right? Just a spark. Like when you take at night when it's cold and you take the sheets and you shake them and you see all these little sparks. Well, those are many tiny lightnings that are occurring. It's static energy that is released. All right, so they generated a spark into this atmosphere, and there was liquid water at the bottom. And what started accumulating over some time, not too much time, was organic compounds. And some of those organic compounds were actually amino acids, of which there are 20 in nature. The amino acids are the building blocks of big, large molecules that do the work, or the workers of life. Proteins, enzymes, proteins are the ones that do the work in life, right? In the cell, cellular respiration, everything, all the metabolism of the cell, the cascades, everything else. So you get amino acid, that's pretty sophisticated, from inorganic compounds. You get amino acids. And now amino acids coming together can form proteins. Proteins can function. And so you can see that we can, with those 7 steps that I listed there, we can get from inorganic compounds and elements from the periodic table, we can get organic compounds, which can come together, form these protocells. Remember the guy in the TED Talk talking about the little protocells coming together and splitting up, and they're doing this spontaneously right in front of our eyes, films, with no intervention of any kind, right? And then eventually the sophistication of an actual cell. Get a cell, a primitive cell will be something bacteria-like, all right, with no organized nucleus. But then eventually we will get like a precursor of our RNA folding upon itself, hairpinning and stuff like that would form a DNA, which is super stable. And then that went on for millions of years because every species that we examine today has DNA. So it means it has to go back to the origin. The first organism on Earth had to have some kind of primitive DNA that has been passed on to all organisms around. And then that's elegant because we can take that DNA from a bacterium, from an orangutan, from a human, and from a fly, and from a pine tree, and look at percentage homology. And we see that there is some percentage homology among all species, which is again fascinating. And then we can, with that, we can do a phylogenetic tree, which is the tree of life, it's the Darwinian thing of evolution, right? Because if we have branches, then those branches are not flying around in space, they're attached to a trunk, and the trunk happens to have a root and so forth. So you see how we can get, at the end of the day, from inorganic material, compounds, elements that are in nature to life. And from a primitive life, again, over millions of years, to multicellular life with eukaryotes. Remember the difference between prokaryotes and eukaryotes? Eo, the prefix eo means true or real. Karios, from the Greek, karion is the nucleus, so a true nucleus, right? In other words, the DNA enclosed in a double membrane of the nucleus, protected and so forth. So once we get eukaryotes, then we can go from unicellular to multicellularity by colonies that form together, and then cellular specialization and all that. We are on the way to multicellularity of the organisms, sophisticated organisms that we see today. It takes millions of years, and that's what we're going to get into, looking at the forces of evolution, the four main forces of evolution, right, that may cause evolution, which is mutation, migration, drift, and selection, natural selection. That's going to be like the second part of the lecture today. The first part, I need to talk about variation. So, questions or comments about the review of the lecture that I didn't give, which I just gave? All right. All right. That's the problem with the professor. You push the button and he just keeps talking. Yep.
I know Darwin, it took a while for Darwin to catch up.
Yeah.
But once the Big Bang came into place, which was what, 50 or 60 years later, more or less, I think.
Yes, more or less. Well, I guess I don't know exactly, but that's researchable. The Lemaitre, probably in the 40s or 50s, 1940s or 50s, so almost 100 years after Darwin.
How well did that kind of like catch on?
The Big Bang?
Yeah. And
Yes.
how about Myers'
Oh,
view on that? Because we talked, I think, at the beginning about him not taking any religion into account, really.
right.
Right?
Thank you.
And this came way after Big Bang, so.
Yes, thank you. Okay, and Meyer was around because Meyer, the sweet thing about Meyer is that he expanded basically the whole 20th century because he was born at the beginning of the 1900s, like 1904 or something like that, and he died in 2005 or something. So he lived over 100 years, and those 100 years was the 20th century. Yeah, of course, when he was a little kid, he had no clue, right? But eventually he grew up, and already in his 20s, he was well into science and being a German. He was very meticulous and so forth. But he expanded the whole thing. And then in the 30s and 40s, he was also part of the synthesis, the modern synthesis that is known, which is evolution and genetics, or genetics and evolution. Genetics as the mechanism for evolution. In fact, we're going to talk about a mutation, which is a random event in principle, as the trigger for the possibility of evolution. So there was a synthesis before we went into space, right? And then there was the Lemaitre theory of the Big Bang, and then what confirmed that was Hubble, the Hubble telescope, which was named after another astronomer, because Hubble was able to measure actually the expanding universe. And so it's backward thinking. In other words, if the universe is expanding and time is moving forward inexorably, with our mind's eye, we can go backwards. So you see, with our physical eyes, we can only see here and now, what's in front of me, and we have a lot of bilateral vision, we have another overlap, so we can see depth of field very well, three-dimensionally, depth of field. We compromise on not being able to see beyond our ears, basically. If I extend my hands both sides and look straight forward, I lose sight of both my hands right around the level of my ears. So it's about 180 degrees more or less, okay? Which means that I see nothing with my physical eyes in the back of my head, all right? And so that's kind of a danger if someone is coming from behind to do an insult. But forward, we have very good vision. It has to do with whether our bipedal, anatomically and physiologically, whether our bipedal stature, that we can walk on two legs only, which frees up the hands for manipulation. I'll get into all that the rest of the semester in detail, looking at the origin of the human species, all right, as a whole. But anyway, back to your question. Yeah, I forgot to mention, my bad, that you can skip chapter 4 of Meyer. All right, we've been going in progress, and for today's chapter 5 on variation. But chapter 4 is basically his refutal of theology and philosophy. Like for him, they're not really contributing anything at best, and at worst, they actually slow down. Philosophy and theology slow down the progress of science. By science, I mean empirical science, that which can be measured, right? Animals and plants and all the biochemistry that's occurring and all that, and the physics, because of essentially where I think that Meyer got stuck is in a fundamentalistic interpretation of philosophy and religion and theology and the Bible specifically. So he's looking at it from this fundamentalistic point of view that take chapter 1 of the book of Genesis. In the beginning, right? Bereshit, and then there was light. God said there was light, so there's the Big Bang anyway. But then there are 7 days, actually 6 days, and on the 7th day, God rested, as if God needed rest, right? So it's metaphorical language. It's a language that is allegorical for a pre-scientific society that had no clue because they didn't have microscopes or telescopes or anything of that. They just knew human experience. But when we get away, and I'm kind of anticipating myself here, but when we get away of interpreting a day as a 24-hour thing, then the alternative is a period of time. And then it matches elegantly with the progression of evolution through those 6 periods of time. The 6th one, the last one, was the human species as such, right? Let us make man in our image and likeness, male and female, we create them. All right, so therefore, chapter 4 of Meyer is basically he's shooting down a philosophy and a theology that we as Catholics do not espouse because it's fundamentalistic, it's a literal interpretation of the Bible. However, to his credit, if you will, or in his defense, that was the standing model of the time. It wasn't really until the end of the 1800s and the beginning of the 1900s that we as Catholics got away from a literal interpretation of the Bible and started looking at source criticism and textual criticism and things like this, looking for the deeper meaning of the passage. So what he's criticizing religion about, that slowed down all the pushback, even Darwin, you mentioned, it took him a while to catch on, but eventually he did catch on, but he didn't publish because he was scared of what was going to happen to them, which is true. And it was the ridicule, and this is in his own time, going back to the 1850s, 1860s, you may have seen the caricatures. For example, you see this kind of stuff, which was all over the place. This is in his own time, right? Oh, we look so alike, the monkey and myself. And so he was, here's a monkey looking, his Darwin looking at a human skull, right? there's a caricature on this one, and the next stage is like the millennials, which is totally technological. And then the next stage after that is a robot, it's a humanoid robot going into the future, but this is going into the past. Anyway, what I want to say was the petite lune, for example, the little crazy one, or the little moon. He knew that this was coming his way because there was essentially a fundamentalistic interpretation of the Bible, and religion had a lot of power and authority in the church. Remember the Galileo trial, right? Which it took John Paul II to exonerate the excommunication 500 years later. Totally exhumacy.
Remember that?
Yeah, well, it was basically under house arrest because Papal States, it was a mix between religion and politics. To this day, the head of the Anglican Church in the UK is the Queen, and there's an official association. Did you see the burial of the Queen? You know, it was all about the church protocol and ceremony and regalia, right? So the Anglican Church is officially the religion of the British Empire, or the UK, right? And so that was the American experiment over 300 years ago, precisely to the land of the free, the expression of all, so that the state would not espouse any particular religion. So you see, there was a lot of cultural background, the context of the time, right? The context of the time was that this was a big no-no. It was going to shake the roots of society at the time. And so it did. But remember, Darwin was pushed to publish. I don't know if I told you the anecdote. Remember, I told you the anecdote. Did we talk about the modern synthesis? We talked about the modern synthesis, right? Okay. So what was the anecdote? A friend of his who was younger, maybe about 10 years, Alfred Wallace. Alfred Wallace was somewhere in Indonesia looking at the same stuff that Darwin was looking at with his trip, with Darwin's trip on the Beagle around the world, mostly the equator, for 5 years, and seeing the variation, right? And so Wallace essentially figured out that evolution could occur by means of natural selection, right? Selective pressure on the survival of the fittest, basically. Survival of the fittest. But remember, fitness is not in terms of the gym, right? Because bacteria have no muscle. Fitness is genetic fitness, the best genes to survive, especially when conditions change. Because if conditions stay the same, we do all right. But look at what happened when the COVID-19 hit the world. A lot of people died. In general, the weaker people died. So that was pure natural selection hammering on the world population for two years. Okay, and we saw it with our own eyes. We lived through it. We're the survivors. Somehow we had those better genes. It's there. So Wallace writes an article about evolution by means of natural selection and sends it to Darwin, the draft, for his opinion. And Darwin says, whoops, I better publish because otherwise this guy, my friend, is going to steal this theory from me. And he's going to get to publish. And therefore, he went ahead and published The Origin of Species, which is his book. I think it was 58, right? 1858. And then, of course, the you-know-what hit the fan and everything happened, which what happened to this day. The other irony of history and the lack of mathematics on the part of Darwin. Remember his cousin Galton, Francis Galton, who was another Brit. He was a polymath, he was a philosopher, he was a physicist. And Galton knew about Darwin. They had talked about his theory and so forth. And Mendel was a contemporary of Darwin, but they never met. Gregor Mendel was doing his speed things here, and he came up with an inheritance factor, because again, Mendel didn't have a microscope, they didn't know about the genes themselves. Those inheritance factors, today we call them genes, right? Embedded within the chromosomes, etc. But the two big theories, independent assortment and all that from Mendel, Mendel published that in basically the statistics and the 3-to-1 ratio, the famous 3-to-1 ratio for dominant recessive in simple Mendelian, which I'm going to talk about today. This is a good segue into the lecture, if I don't. So Mendelian genetics, Mendel published in an obscure little mathematical journal his results from the 3-to-1 theory, all right, the 3-to-1 results. Galton saw that article as a mathematician. He was staying up with the science of the time and the articles of the time, the publications. And Galton showed that article to his cousin Darwin, saying, look, this may help this may have something to do with your theory of evolution. The anecdote goes that Darwin put it aside because he couldn't understand the math. So it was all qualitative. In fact, if you read The Origin of Species, which I highly recommend to you, is both The Origin of Species by Means of Natural Selection, you will find in that book, first of all, not a single reference. No credible article today, no peer-reviewed journal or book or press is going to publish anything from you if you don't put references in it, right? So that's the first thing to do. Sometimes these articles is so cumbersome because practically every sentence is referenced, and you have 1, 2, or 3 references for each sentence. It's overloaded. Anyway, that's one. Number two, there's no math in Darwin's stuff. It's not quantitative at all. It's all qualitative. You know, it's the color of the bird. It's the variation on the tone of the feathers, and it's all qualitative. Longer length of the arm, shorter length of the fingers, and so forth. And so, but he got it out there finally. Anyway, that's a good little segue. Less other questions or comments. So, with regards to my criticism of Meyer's criticism of religion, faith, and philosophy and theology, is that he was also a product of his time, and the culture of the time was basically fundamentalistic. And therefore, he's rejecting a type of science, faith, philosophy, and religion, and theology that we also reject as Catholics. Okay. But the reality is that that kind of fundamentalistic approach did push back on the theory of evolution. Now, today, the lecture is actually 2 parts. The first part is on variational evolution, which is Darwin's one of his main points in his book and his theory. Is he doesn't really like to talk about species. He likes to talk about variants, all right? Variants. And so, for example, we look at cats today. We could look at a black cat, we look at a calico cat, we look at striated cat, we look at different patterns of cats, right? But they're all cats. So it's one species. But he would be focusing on the variations, on the variants. And then he'd be looking at, actually, some cats actually are born with 6 toes. Well, is there an evolutionary advantage to that, or a disadvantage? Or is it just innocuous? That kind of thing. So he's looking at variation, and he's looking at ranges then, ranges. So when that's what we're going to look at, population ranges, and then polymorphisms, which are characteristics. We have to get into genetics finally, a little more detail, quantitative traits, and then Darwinian evolution. And with Darwinian evolution, in the second part of the lecture, we'll actually look at the 4 main forces of evolution, like I've mentioned before, which is mutation, migration, drift, and selection. Okay. Forward. Variation can happen at these 4 levels. Morphologically, physiologically, molecularly, and behaviorally also. In other words, 2 individuals of the same species, obviously, that are morphologically alike and physiologically and molecularly may still behave in a different way. So that's a variation. Probably the behavioral one is the hardest to understand. And I want to mention that all living organisms have behavior. We're used to animals having behavior, right? Running away from fire, being attracted by a mate, etc., etc. But plants also have behavior. Oh, how can plants behave? Well, a branch and a twig and a leaf, does it go toward the sun or against the sun? A branch, a twig, and a leaf, does it move in the tree, in place, Does it move locally toward the sun or against the sun? Toward the sun. That's called a tropism. It's a movement. And it's called a phototropism because the stimulus is photons or light particles, right? So that's a positive phototropism, and that's a behavior. Now, what about the roots? Do the roots go toward the light or against the light? Against. So you have a plant, you have an orchid at home next to your window. The stem where the flowers are going to come out, which are adapted leaves, super specialized adapted leaves that have turned into flower, into a different reproductive structure. That twig is going to go toward the window, but the roots underneath the orchid are going to go against the window, toward the darkness of the room. Okay, so that's a negative phototropism. Again, a normal tree or normal plant planted in the ground, the roots have a negative phototropism, but they have a positive geotropism. What do I mean by positive geotropism? What's the stimulus of a positive geotropism? Geo has to do with Gaia, which is the earth, right? Is that would be hydro. This is geo. Again, the most obvious force that we take for granted, keeps us grounded so we don't fly out. Gravity, geogravity, right? So gravity is pulling on everything that's on Earth and in Earth and around Earth, including the roots. So the roots are reacting positively toward the pull of gravity. And it's pulling them toward the center of the Earth. If a tree could get a root to the center of the Earth, it would. But it can't. Okay, so that's a positive geotropism. Whereas the branches, what kind of geotropism do they have? Do they have a geotropism? Yes, they do. But it's a negative geotropism because the roots are moving against. The trunk is even more obvious. The trunk of the tree or the plant has a negative geotropism. It is reacting against the pull of gravity. So plants also have behavior. And bacteria also have behavior. And so if bacteria have behavior, then everything, even fungi also have behavior. They're attracted to where the dead carcass is, right? Fungi, and will continue to grow around it and so forth and digest it. So that's behavior. All right. So these four levels, the ones that are most observable, of course, are the morphology or the shape of the thing, the structure of the thing. Physiology is not as observable because it's mostly internal. This could be at the cellular level, at the tissue or organ level, or at the level of the whole system. For example, digestion occurs in the digestive system, and that's a physiological thing. Then molecular, obviously, at the molecular level, and behavioral. So the ones that we can normally observe and measure without much instrumentation, which two of these four? Yeah? And behavioral. Morphological and behavioral, we can see, we can hear, we can smell, etc. We can hear birds singing to each other and so forth. But physiology and molecular, generally we need instrumentation to measure it, right? But we can measure it. So variation occurs. I'm talking about within two individuals of the same species who are very closely related. They may be siblings, right? They may be progeny of the same parents. So their DNA is very similar, but there's going to be variation, even at that level.
He was mentioning in the book, identical twins not being
100%.
totally identical.
Correct.
I thought that at the genetic level, identical twins are identical. But I guess what he's talking about is the behavioral part.
Well, not only that, that's exactly what I'm going to do today with these two ladies that I'm going to introduce to you. I'm going to look at, because if we find variation with identical twins, then there's obviously variation in everyone else.
You're talking the variations in the identical twins at the genetic level, you're just talking about those polymorphisms?
Yeah,
Like
the random mutations. Yeah.
That really probably don't mean much because
They may or may not. Most of them are in the junk region, so they're neutral and all that. You're kind of anticipated today's lecture, but that's where I'm going. Exactly. No, that's exactly what I'm doing. That's why you work with DNA, right? It's your daily bread. Yeah, but okay, so basically this is the drift. If I can make the case, if we can make the case for variation within identical twins, for variation between them, at those 4 levels, and I'm going to prove to you just with a photograph, variation at those 4 levels of this identical twins, then there's variation in every other 2 individuals of the same species on Earth, right? Now, when we talk about twins, we have to make the distinction between the dizygotic and the monozygotic, because identical are the monozygotic. Okay. Now, dizygotic, they come from 2 sperm that fertilize 2 eggs, which the woman happened to ovulate 2 eggs in that cycle. That's as simple as that. Or 3 or 4 or 9. I think what's the record? I think 9 non-lactose, a woman who actually gestated and gave birth to 9 twins, but they're not identical. With 9 eggs. Yes, it's a record. It's a record on Earth, 9. I forget now if they were identical or not, but there were 9. She's surrounded with this 9 little tiny, all premature, right? Or no, but low weight birth. Right. So anyway, 2 or more eggs that are around and millions of sperm, every egg is going to get fertilized in principle, and those are dizygotic twins. Now, because it's a sperm that determines the sex, right? Either an X sperm or a Y sperm, then dizygotic twins or fraternal twins can be either boy, girl, or boy, because they come from 2 separate eggs, right? Okay, so we're clear on the genetics and all that. Now, monozygotic twins, zygote is the first cell of any individual in sexual reproduction, including humans, and mono is single or 1. So we have 1 egg fertilized by 1 sperm. That individual is going to be either a male or female, depending if the sperm was what? X or Y. End of story. Now, so the zygote within 24 hours is going to split into 2, undergo mitosis. Cellular division is from the zygote forward. It's all mitosis, your tosis, everybody's tosis until the grave and after the grave. Okay, so from then on, it's just mitosis, simple cell division, 1 into 2. What's happening? We're duplicating the number of cells, right? So they may actually share placenta, etc., different stages, but depending on when the split happens, the point is that at some point within 2 weeks of fertilization, of gestation, we're done with fertilization. Now we have a fertilized egg, we have already a zygote in the human and non-human, in mammals. Within 2 weeks, that little embryo can actually split, and those will be identical twins. And in fact, they can split once and then they can split again, and they will be quadruplets, right?
The consensus on beginning at fertilization is because at fertilization we have that unique DNA for that unique individual.
We have a diploid cell, right? We have that's actually now you're going into next semester's course, which is the beginning of life. But that's basically the bottom line argument is that the egg is a gamete, the sperm is a gamete, which means they're haploid, they don't have a complement, they don't have a full complement of chromosomes, they don't have a single number. 23 chromosomes on the egg, 23 chromosomes on the sperm for the human. When they come together, now we have 23 pairs, which makes a diploid cell. That diploid cell is a new individual of that species. You know, it has 50% of the father and 50% of the mother, but that individual is a new individual genetically. Going forward, left alone in its own place, 9 months later, a baby will come out. And so it's all biological. I don't need to mention God, faith, religion, or theology at all. It's all biological.
A universal consensus on science.
Yes, absolutely. More and more so. It's so obvious. And it's not only that, it's not even unique to us. It's all mammals, and in fact, all animals that reproduce, and even plants that reproduce sexually. What's the equivalent of sperm in the plants? The yellow stuff that is dirtying all the cars after the car wash, 5 minutes later, it's covered with yellow. Pollen. Each pollen grain, remember, has 2 sperm nuclei inside. So the flower, which is the reproductive organ of the plant, inside the center, the pistil, the eggs are there, the ova are there. And the pollen will fertilize those ova, and you end up with a zygote. So any honest biology textbook from high school, you open it up in plant reproduction, and the first cell is a zygote. So it's universal, right? Because DNA is the universal molecule of inheritance. That's the argument there. Anything else is going to be, with all due respect, ideology. Ideology that is defective is not true to the science. So we're clear with identical twins, therefore they have to be of the same sex, either male or female both. Let's say that the split happened and only 2 siblings, right, instead of 4 or 8. Keep it simple. So these are Claire and Laura Newing. These are actually human beings who exist, and they are identical twins, monozygotic, and you can see they look very, very, very similar, but they're not identical. And I'm going to prove to you this morphologically, physiologically, molecularly, and behaviorally from this one photograph how they're not identical. Well, they're not 100% identical. They may be 99.99% identical. From this one photograph, let's examine it scientifically, scrutinize with critical thinking. All right, so here they are, Claire and Laura. Let's look at morphology. All right, so morphology is the shape or the structure of a body part or the whole body, the whole organism. I cropped this photograph on purpose on the top to touch the head of Claire. And this line over here, what happened to the head of Laura? Exactly. So we can safely assume that whose femur, what determines mostly the height of the human? What bone? The femur is the longest bone in the body. And generally, that determines more or less the height, right? So it's safe to assume that Claire's femurs are slightly, maybe an inch longer than Laura's femurs. That's a structural morphological thing, okay? Now, of course, I could be contradicted because maybe Claire is wearing high heels or higher heels than Laura. We don't know. The photo doesn't show the feet, but it's an assumption, okay? Anyway, that's morphological. Physiological. Let's look at a close-up of the freckles of their faces. Do they have the same amount of freckles? Of course, if I make it larger, what happens is that it starts pixelating and the freckles start disappearing. But let's make it a little larger for now, just like that. And just at face value, which one seems to have more freckles? The one on the right, who is Laura, okay? Laura seems to have more freckles. Anyway, so that would be a physiological thing, right? Because what determines the freckle? It is a protein that's inside the cells that is called melanin. And the melanin expands or contracts, reacting to the light, to the photons. And so if Laura has more freckles, we can simply assume that overall, quantitatively, Laura, at least Laura's face, has more melanin than Claire, and that's a physiological thing. Okay, we're just having fun with this. How about at the molecular level? Well, I gave it away because the physiological would be actually be the freckle as a whole, but the molecular is the melanin, which is an actual protein in the skin. Okay, so that's molecular. It could be also that one likes sushi and the other one doesn't like sushi, which is a behavioral, but also a molecular thing, because maybe one is allergic to shrimp and the other one is not. I don't know. But that's too speculative. Behavioral. How can we determine behavioral from this photo? Yes? Yes? Very good. Yeah, it's true. It's a little more subtle to me. But what's a behavior that, again, we take for granted? because we're normal human beings and we go out into the street normally naked. We dress up. And how are they dressed? Differently. So they have different tastes for dress. So they dress differently, you know, kind of similar with a little bow tie thing here, but they have different blouses. And so that's a behavioral thing. It's just choice. They like one blouse better than the other, right? Also, the hands, it's interesting. One is holding the paper on the right hand, the other one's holding it on the left hand. But they both have a ring on the middle finger. So there's behavioral. Okay. Anyway, that's my point about this. That if two identical individuals that are monozygotic and have slight variations between them, then everybody else, any two other individuals of the same species are going to have variation, let alone different species.
The case of a dominant genetic hereditary, let's just say cancer mutation, like the BRCA gene.
Yep.
If you have identical twins and one tests positive, can we assume the other one tests positive too?
Most likely. So, you know, yeah. So, you know that when we go into medicine, we're going into a statistical model, a probabilistic model, all right? And we're forever at this quandary of going back and forth between a probabilistic model and a deterministic model. What I mean by a deterministic model, that we're not probabilities. When I go to the doctor and the doctor says, well, you have a 50% chance of having a heart attack because your dad had a heart attack and his dad had a heart attack and died from it, you know, then you'll have a higher risk. It's a probabilistic thing, but I'm not a probability. I'm a determined individual human being. So going back and forth like that, when you say the chances of, that's probabilistic language, that's statistical language. So I would say yes, 99 point something percent that the identical twin will also have the BRCA2 or BRCA1, right? But unless I do the test on this individual determined, either individual A or B, I will not know for sure. And even that test has a margin of error. So it's the back and forth. Okay? Yeah. Now keep in mind that because there's one fertilization here occurring by one sperm and one egg, that original DNA, the diploid DNA of that zygote, is one and it's identical to itself. All right. But then going forward, after already at the first mitosis, there can already be some chance mutation. Now, no, because 90% or 97% of the DNA is non-coding, right? Then chances are that the mutation will fall in a non-coding region. The chance 97%, only 3% chance that a mutation, a random mutation, will fall within the coding region, within an exon that actually expresses for a protein or a polypeptide, which could then likely damage that polypeptide, especially if it happens on the active region, on the active side of the protein. But if it happens on the non-active side of the protein, it's innocuous.
Rare disease causing mutations outside of the exons, correct?
Yes, yes.
Much rarer.
Yes.
You're not picking anything up on
That's right. That's why when we talk about junk DNA, you know, that 97%, which now since statistics going 98 or 99%, when we take into account the introns also, it's not really junk in that sense because it may not have coding as such. It may not code for an amino acid, right? There may not be a triplet that actually codes for an amino acid in the genetic code, but it has some other function because it's still there. And it could be at the higher order level of the DNA of the folding, the three-dimensional folding, the chromatin, how it's all wound up tightly, and how does the replication machinery get in there, or the transcription machinery get in there to read a little section, right? It has to do with the three-dimensional overall. So it could be that the junk DNA has that higher order function of folding the chromosome in a particular way.
It still appears to us as junk DNA, but it in fact, as time evolves,
Yes. Exactly. So we have to qualify junk in the sense that there's no coding region. Right? There are no triplets
Right.
of the bases that actually codes for one of the 20 amino acids. And that has been deciphered already, the genetic code. Okay, so again, the picture says 1,000 words. Yes, exactly. But remember, it's the RNA, so instead of T, it's the U and all that. This is what I'm talking about. This guy here. Where you have the triplets, right? And oops, I'm sorry, I forgot to minimize the little tile. That's going to be in the first part of the lecture. Right. Each one of these triplets codes for an amino acid, phenylalanine, leucine, isoleucine, methionine, valine, alanine, tyrosine, etc., proline, right? The 20 amino acids. And then you have the triplet code, which is redundant and it wobbles. I'll talk about that in more detail. But yes, there could be, so coding region specifically is that 3% that we talk about, which now is even smaller to maybe 2 or 1%.
Really, of coding?
Yeah.
We're gonna talk about the exon, you're talking outside of the exon.
No,
In
exon, strictly
Oh,
exon, strictly exon. And then the other aspect also keep in mind is the gene and its environment, right? Yeah, it's really tiny, amazing.
Only 2% or the errors
Yeah.
can happen.
Exactly. So it's even safer.
Yeah.
It's not only inside the cell, it's inside the nucleus, and it's also bunched up very tightly, a compaction of about 10,000 times compaction to get the chromatin inside the nucleus of a cell, right? And then to boot, only 1%, maybe 2% of that whole string actually codes for protein or for polypeptides. So it's, again, for lack of a better word, we keep talking about the elegance of nature, right? Okay, folks, back to the twins. We can agree, hopefully, that monozygotic twins can also have variation at the morphological, physiological, molecular, and behavioral level. So the standard out there and in here and everywhere that there's life is variation. Why? The point about variation is that selection is going to act on variation. Precisely. So it's a selecting out. For example, in Claire and Laura lived in equatorial Africa or equatorial anywhere in the world. Which one is more likely to get skin cancer? The one with the fewer freckles. On average, see? So that's a selection. So if they live in equatorial Earth, Laura, I think it's Laura, sorry. Yeah, Laura will be more likely to survive skin cancer. But if they live in the in Northern Europe or something like that, which probably that's where they come from because of the blonde hair, then the freckles are less relevant, right? But if they had more hair in their body, for example, then cold, the one who had more hair would be more likely to survive a cold winter. Just kind of exaggerating. But you see my point about the variation. Variation is the game of survival because selection acts on variation, right? Then we can put variation on a range of stronger or weaker. Variation. Here are some examples of variation. How many lions in this photo? Yes, there's one back here. Don't miss it. There's a cub back here. Of course, the fur is camouflage, right? It blends with the environment, but there's one little guy back here. There's Mama Lion there with the 3 cubs. I think this is called a pride. Anyway, is there variation? Look at the cubs. We assume that they come from the same mama, so these are siblings, and they look very much alike. I would say, they're identical. Check out this ear. There's a black spot, right? This ear has no black spot that we can tell. So that's a variation. It's subtle, but it's real. Let's say that these are males. And a female lioness selects, she has a preference for dark spot ears. So this little guy's gonna win out because he doesn't have the dark spot ears. So I'm just saying, you see, selection may be subtle or variation may be subtle, but real. And that's just on the side that we can see, not to mention the smells. Maybe one guy is more smelly than the other, or maybe the female prefers more smelly or less smelly, I don't know. But that's another one. It's on the physiological and it's on the molecular, right? Because of the musk or the smell of the animal. In fact, the male is usually looking for the smell of the female to determine when she's ovulating. So all that variation is very clearly out there. Tulips, they're all the same species, and you can certainly see a range of from yellow to red going through orange, right? And there's variation very obviously there. And all the children of the world show tremendous variation within one species of Homo sapiens. So variation is the name of the game, and selection acts on variation. Now, I want to introduce the reality of polymorphism. Here is Canis lupus familiaris, which you see the dog is a subspecies of the wolf. Domesticated, just about the only animal we have actually managed to domesticate of the hundreds of thousands of species that are out there. The zebra, for example, even the dog is, even the horse is kind of domesticated, but the horse actually, the wild horse has to be domesticated before, otherwise it remains wild, right? Wild horse. The zebra, they say, that for centuries, for millennia, humans have tried to domesticate zebra because they're very strong and so forth. They have the quality, they have speed and strength, which is a combination. And African tribes for millennia have tried to domesticate the zebra, and they get kicked away because the kick of a zebra can kill a lion. And the zebra, they say, has a nasty disposition. They also spit, and they're real nasty. You can't domesticate a zebra. Anyway, but the dog got domesticated somehow, and here it is. It's all one species. And look at all the different breeds that we have of dogs, right? That's all variation. These guys are all varieties of the same species, okay? Which means that in principle, if they can work out the mechanics of it, they can interbreed, right? They can interbreed. From a German Shepherd to a Chihuahua, whatever this is, in principle, a male and female of those two different breeds can interbreed and produce not a hybrid, but a fertile offspring, okay, which is the definition of species. So they all belong to the same species. But of course, we know that some breeds have become so awkward that they cannot really interbreed mechanically because the breed has become too degenerate. But in principle, they could. So they belong to one species, and that's what we call polymorphism, the range. And there are several ranges. What is the most obvious range? There are two very obvious ranges here, three actually. But the most obvious range, at least just looking at the picture as face value, and even the way the dogs are arranged on the photo. It's a morphological thing. The size. There's a range of sizes, right? From the smallest to the largest. And most dogs, like most animals and plants and humans, are going to range about a mean. So we're into the probabilistic world here, okay? There's going to be a normal, many ranges in nature are normalized, are normal, not normalized, but normal. They're normal distributions, right? The bell-shaped curve presented by Mr. Gauss, or the Gaussian curve. Most individuals within the species hover around the mean within 1 degree of freedom and all that, right? So most dogs are going to be a mid-range size. Mid-range, then you get a few who are smaller, a few who are taller and or bigger, and then very few who are very tiny, like the little Chihuahua, and very few who are huge, like the Mastiffs and all those huge dogs, etc., right? But those are fewer, not number of individuals, but number of breeds. And that's how we get the distribution. The same with the human. The average height for men about 5.9 or 5.8, whatever it is. For women, 5.8. There's a slight dimorphism of size, about a couple of inches from the woman, the average woman to the average man. But we have to look at what? We have to stay within what? If we're going to measure, let's say, height of men and women of average, we have to stay within an age range, because obviously a 5-year-old is not fully developed yet, so they're going to be shorter just by the age range. So if we say adults, okay, that's one thing. Or we say 16-year-olds, or we say 5-year-olds, or newborns, right? Each age will have its own range, but generally it's normal. And many distributions in nature are normal, okay? And so there it is. That's what would be my polymorphism. Each individual on the range is a morph or a shape. And that's with regards to size. We could also look at length of hair, for example, or amount of hair on the body of the dog. That's another one, and so on and so forth. So there are ranges all over the place. We could look at the ears, whether the ears are fully pointing or half pointing or droopy, and we can develop a range there of ear flaps, if they're long or tall or short. See, so that's what we mean by polymorphism. The range of characteristics that can be, again, morphological, physiological, molecular, or behavioral within the same species, because all of this is pointing to variation. It could be going forward that some breeds, when they go into the wild, have a survival advantage versus other breeds. Okay? And that would be selection acting on that individual. All right. Now, many distributions in nature are normal, but some are not normalized. Okay, they're skewed, either positive or negative. Now, this is a little counterintuitive, because you notice that, for example, for a positive skew, the bulk or the largest number of individuals is to the left. So why is this called a positive distribution if it's to the left? You would think it's negative, right? Because they're the smaller individuals. It's called positive because the skew is to the right. The skew is to the right, okay? So that's why it's called. So the positive is to the skew. The skew is a noun. The positive is the adjective, which is qualifying the noun. And therefore, the sign, if you will, is on the skew, is not on the most. Fewer are skewed, and that makes it positive. Where fewer are not skewed, in other words, most are on the right side of the curve, then that's called negative, because the skew itself is to the left. Okay, so it's a little counterintuitive. People tend to call this a positive and this a negative, but no, because it's on the skew. In other words, it's leaning toward where? The tail. It's also known as the tail, okay? The long tail. These butterflies are known as heliconians, and they're all from the same group of butterflies. They're related to the dragonfly, which you can see the dragonfly is not a butterfly, but it has similar kind of wings than the heliconians, right? In fact, there's one of these heliconians that is called a glass heliconian, and they're transparent. Butterflies that are transparent. And have you seen these glass butterflies? They're fun. They're not on the East Coast, they're on the West Coast. They're fun to watch. Oh, no, that's not what I mean. These guys, these are real life. Look at that, you can see through them. They're not made out of glass, they're made out of organic molecules, but they don't have scales, or they have very few scales, or maybe even transparent scales. That's what makes the butterfly wing pattern. There's scales. And that's why they call it Lepidoptera, because lepido is a scale. butterfly scale. This one needs a microscope or at least a stereoscope to see them, but look at that pattern. This is actually what produces the pattern of the wings. See, they're like little tiles. Amazing, but they're tiny, microscopic. Here they're rubbed off. Okay, so the little scales, it's just fascinating when you see one of these. You don't realize you're looking at a microscopic view of a butterfly wing. And the moths have the same, only that the tips here are flared out and hair-like structures, so it's a more fuzzy looking color or pattern. Okay, the distribution of men and women in the human population, and this happens with many species, is known as a bimodal distribution. This is another type of distribution. So we've seen normal distribution, we've seen skewed left and right, and now bimodal. Bimodal distribution is the size, the tallness, all right? Because, let me see if I can make this a little bigger for a moment. The average, so first of all, I have to qualify, I mentioned earlier, let's say for adults, right? For average adult humans, we know that the average adult man is a little taller than the woman. This is measured in feet, the scale. So 5.2 feet is the average height of women across the 4 billion, well, less the youth, all right? So let's say these are adults. So this number, by the way, is very robust because it's made up of millions of observations, right? And 5.2.5 and 5.7.5 for the man, for the male. So we have 1, 2, 3, 4, about 5 inches. Yeah, this is across the world, right? So this would include people who are very short in general, like Asian people are shorter, African people tend to be taller, and so forth. So this is a world average, right? As far as I know, I think it's a world average. I can't really tell you for sure right now where I pulled it out of, but yeah, it's about a 5-inch difference, which is pretty large, pretty significant, right? This is what we call a moderate dimorphism, DI. DI for two morphs, a moderate dimorphism between men and women, the average height of men and women throughout the world. Some species have moderate dimorphism, other species have extreme dimorphism, like for example, the male and the female gorillas. The female gorilla looks kind of like a chimpanzee, about 4 feet tall, exact. For example, the skeleton at the entrance over there in the hallway, that's a chimpanzee skeleton, right? Chimpanzees, by the way, they don't have practically any dimorphism between the male and the female. They're about the same size and look, at least from us looking at them. But the male gorilla is, I mean, almost twice the size of the female gorilla, right? It's huge and much more robust and so on. So that's an extreme dimorphism there. It's all according to the evolution of the species, right, and the selective process. But that's a dimorphism for humans with regards to the height. So this, just using this illustration as a bimodal, because to talk about the average height of humans, both together, well, it would be 5.5. You see, but because we're either male or female, physiologically, anatomically, etc., then we actually have a dimorphic distribution, or I've been saying bimodal distribution of tallness. Okay, let's move forward. This is the whole thing. Okay, so here is just a little slide on going back and forth between the deterministic model and the probabilistic model, because we use probability a lot in nature to study morphs and to study polymorphic traits, okay? And so First, just some very basic skills that need to be developed by anyone who aspires to be a scientist or even just a normal human being, to be able to translate between fraction, decimal, and percentage. This is something that we should be able to do in our sleeps. And I can tell you, many people just don't know how to do this. They get stuck. And so they have been deprived. I tell those people, go back to your high school and your elementary school and ask for a refund, because you have been gypped, all right, to translate a fraction to a decimal to a percentage. The simplest one to do intuitively is a half, which is 0.5 or 50%, right? But then 1/3, people get stuck already, because what's 1/3 in decimal? Is it 0.3 only, 0.30? No, it's 0.33 progressive. The technical word is progressive, and it's 0.3333 to the infinite until the cows come home. And so how do you write that? There's a notation to write that, which is 0.33 and a little line on top of it, right? A little horizontal line on top. That means progressive. That means that number, that sequence of numbers keeps repeating indefinitely until the pen falls off the finger, off the hand. And in percentage, is it just 33%? No, it's 33.33 progressive. Because in percentage, you're just dividing by 100. So you're just moving that decimal point over 2 and adding the notation of percentage, okay? So 1/3 is 33.33 progressive percentage, right? Not to mention 1/27th. Exactly. So you see, it depends what we're using the number for. And in general, the British system from the British Empire deals with fractions, feet, and Fahrenheit, which is non-decimal at all. But then the metric system is decimal and is standard, but sometimes the metric system can be a little awkward for a fraction. In other words, the number can be more easily represented in a fraction way as opposed to a decimal or percentage, but percentage is usually the language for probability or statistics. So we have to get used to dividing by 100, okay? Then the mean, which is there are several different types of mean. The mean that I'm talking about is the arithmetic mean or the average, which is what's the mean of 2 and 4? 3, because you add the 2 and you divide by the number of integers, which is 2, and that 6 into 2 gives 3, all right? So that's the mean. So it's a sum of the individual numbers or individual digits divided by the number of numbers or the number of digits. The range is all of the individual members or observations within that population. So now I'm introducing another technical term, which is population, which any number of individuals of the same species in a particular region. We talked about population before, and we're going to pick up that terminology again, right? The variant is any individual within that specific population. So these are just basic terminology, the mean, the range, and the variant when we're talking about variation. Now, genetics. 3 key little words here: the genotype, the phenotype, and the genome, right? So the genotype is the genetic makeup, the actual DNA sequence within my individual cells, within the nucleus of my individual cells. That's the genotype. The phenotype is the observable, the expression of the genotype. So the genotype is opaque, it's not seen to us. At least by the naked eye, we need to use some kind of instrumentation and a sequencer machine to sequence the DNA, et cetera, et cetera. But the genotype is opaque, we don't see it. What we see is the expression of the genotype, which is known as the phenotype. or characteristics. And those characteristics, guess what? They're either morphological, physiological, molecular, or behavioral. So those are the characteristics that derive from the genotype. Those are the phenotype or the phenotypic characteristics. So what was Mendel observing in his pea plants? The genotype or the phenotype? Absolutely phenotype. Tell me a couple of those. One phenotype, and it was an all or none characteristic that he was looking at, right? The 7 of them. So what's one of those pairs on the pea plant, or the pea seed, or the pea pod? The color of the pea, green or yellow. The shape, smooth or rugged or corrugated. The actual pea pod, whether smooth, inflated, or contoured, right? Then the plant itself, either tall or short, so there was no polymorphism. In Mendelian genetics, there's no polymorphism. It's rather all or none. The phenotype are 2 alternatives. We either have white desk or black desk. There's no gray desk around, right? Very rare to find Mendelian genetics in nature. Most of it is polymorphic. It's a range of expressions. Okay? What's the first level of phenotype? If the phenotype is the expression of the genotype, what's the very first level of the phenotype? Of expression, level of expression. What's the first phenotype? Hints. It's molecular. It has to do with the central dogma in biology, molecular biology, review of the central dogma, transcription, translation. Those are the functions. What are the structures? We begin with the genotype in the nucleus, which is what? Structurally, molecularly? DNA, the most obvious thing, right? So that's the genotype, that's the structure, the DNA, right? What's the central dogma? DNA to RNA to protein, right? So from DNA to RNA, transcription, from RNA to protein, translation, right? Or protein synthesis. Again, it's so basic. I'm going back to elementary school here with a good science course in elementary school. This was already covered, all right? The central dogma of molecular biology. DNA to RNA to protein. Because the DNA never leaves the nucleus, and the proteins are in the cytoplasm doing the gazillion work that needs to be done on a daily basis, on a minute-by-minute basis, right? So how does that happen? The how? It's a mechanism. Transcription, translation. All right. So what's the first level of phenotype, of expression of the genotype, which is the DNA? The first level of expression is translation, protein synthesis, the polypeptide. So the first phenotype is the polypeptide that is going to form a functional protein in the cell. Is that molecular or what? Yes. Yes. And it's also physiological. When you take that cell, you bump it up to tissue, you bump it up to an organ like the stomach, for example. Now you get digestive enzymes. So the first level of phenotype is the protein, the functional protein, or the polypeptide that makes the functional protein. Make sense? So imagine that's at the molecular level, and you can expand it all the way up. You go through the levels of the hierarchy of organization, the levels of organization in organisms. Cells with similar functions from tissues, tissues with similar function from organs, organs with similar functions from systems, and systems with different functions integrated from the organism. But it all begins with the genotype, which is opaque. We don't see, but we see the expression of the genotype or not. The reason why I mentioned this is because not all genotype is going to express all the time, right? So not all proteins are being made all the time in every cell.
So this
Yeah.
translational, because I know there's like a translational research science Did you all really focus on this initial,
Yes and no. Yeah,
you know,
this, the central dogma and the mechanism, yes and no, okay. This is an opportunity to introduce another word that we use in science and many places, is univocal. We say that translation is not univocal. All right, let me get my whiteboard up here for a moment. Give me a new page and let's make it big. And let's start spelling some words here that have been thrown out without spelling for you. Univocal. All right. This is the added value of the program and the course, that you learn a little Latin and Greek along the way. So compound words, vocal comes from voice, from voce, from saying, from word. Uni is one, single, all right? Many words are not univocal. They're not univocal, meaning that they have different meanings depending in context. So again, the frame of reference, the context. When I say translation in molecular biology, I'm talking about protein synthesis. In other words, the mRNA being translated into a protein, all right? That's translation within the context of molecular biology. Translational research is research that occurs at a university or a lab and is translated into something that can be marketable, like pharma, all right? That's translational research. So a number of universities, especially the bigger universities that have bigger budgets and therefore they can afford big time research, they have what is known as industrial parks embedded within the campus of the university or sometimes off campus. But an industrial park is a hybrid, is a relationship, it's a partnership between the university and some industry, like pharma. The pharma would pay the money, the university will provide by way of grants or donations or appropriations, whatever. And the university will provide what? The researcher, the infrastructure, the lab that's needed, procuring the equipment to do the research, the thinking process, the graduate students or undergrad, whatever, you know. And the pharma is providing the money for the thing to get done, for the trial on pharmaceuticals, which eventually will reach clinical trials, so on and so forth, and will be marketable. And then that pharma will have the patent on that medication for X number of years before they have to release the patent, and then it becomes public domain, and then other pharmaceutical companies can copy it, right? But for the next number of years, because that Bayer or Pfizer or whatever it was, Russell Uclaf, which is the one that makes RU486, the abortion pill, right? Russell Uclaf, they developed that mifepristone for that specific thing. They have a patent on it for X number of years, and then after they made their millions, they have to release the patent for others, and then other copycats will copy it. You know, that's the business world. But these industrial parts, that's translational research that can be marketable. It's a potential for that may be marketable.
It might be marketable.
That might be
It's or laboratory related or
all kinds of engineering you can imagine. I mean, all kinds of stuff. Microwave, NASA is big into that. Yeah, so I mean, right away the mind goes into 1,000 directions of creativity, right?
Right.
And by the way, it's not tied to science only. It's also tied to the arts. It's tied to performing arts. It's tied to colors and plastic materials. For example, the fashion industry is very concerned now about global warming and pollution, because it's one of the biggest impacts on nature is the microfibers that are coming off of synthetic products of clothing in the wash that goes into the sewer, into the ocean. So one of the biggest offenders of pollution is fashion, especially with synthetic materials that last. These fibers, these polyesters last. Well, they haven't been around for centuries, but as long as they've been around, they've still been around. Okay, so they don't really break down to the molecular level where nature can reabsorb them and recycle them. in contrast with the natural fibers like cotton or even silk and stuff like that, that is made from organic compound. Okay, so all kinds of research going on today in all fields, imaginable and unimaginable. As long as there's a profit to be made, the human will investigate it. Okay, folks online, are you still with us? Any questions or comments? Jump right in, okay? All right, so the genome is a complete genotype, if you will, of the entire species. That's the genome. So this is at the, if you want, the genome is at the genetic level, right? So when we talk about the human genome, that includes the 8 billion of us, whereas the genotype, each one of us will have our own genotype. So it's just the level of magnitude. Basically, in general, the genome is more at the population level of that species, but the genotype is at the individual level of that species, of that population. Now, further into genetics, just very quickly, because you're all familiar with this, it happens that all of you have a scientific background, and therefore, we start with the DNA sequence here, with the 4 bases, right? A, T, C, G. So this is DNA because it's got T instead of U, which would make it RNA. And this is a flattened out double helix. You can see the double helix here, which is also known as a twisted ladder. That's the sequence. And it's a complementary sequence. What do I mean by complementary? If I give you one strand, you can decipher the other strand, the complementary strand. Why? Because the bases always pair up in a particular way. A goes with T, and C goes with G. So if you think of the capital letters, a very easy way to remember it, a mnemonic, mental memory is, in capital letters, the straight letters go together and the curved letters go together. Right? So A and T straight, C and G curved, right? So that's an easy way to remember, but that's what we call complementary strands. And that in fact happens in replication because the replication machinery gets in here and does the leading strand and then does the lagging strand individually. And that's how you end up with double. In replication, right? When the DNA of each cell duplicates, replicate or duplicate is the same thing, before mitosis, before the cell splits, so that the daughter cells end up with the same number of chromosomes as the original mother cell. Very important replication to occur before cell division, before duplication, before mitosis. Anyway, we take that double helix and we wind it up, and we start winding it up within itself. So the first level of coiling is a double helix, right? That's the first level of coil. It's a coil, right? It's a spring, it's a twist, right? That double helix is going to call upon a little ball of proteins that is 8 proteins or 4 pairs of proteins, and they happen to be called histone proteins. Histone proteins. Histone proteins are very conserved, meaning that the sequence of the histone proteins are very similar from us to the fly, to a grasshopper, to a pine tree. The histone sequence is very similar. Histone A, histone 2, histone 3, histone 4, all right, A and B. And so there are 4 pairs that come together in a little octave, little ball, and the DNA winds around that ball like the string winding around a yo-yo, right? Almost 2 times, 1.7 times, too much detail, don't worry about it, but almost 2 times winds around and then shoots out again and winds around another histone ball and keeps going like that. This structure together, the DNA and the histone complex together, is called a nucleosome. Nucleosome. And the nucleosome, again, compound word, some, soma, body, nucleo, in the nucleus. The nucleosome is the functional unit of DNA. The nucleosome is the functional unit of DNA. When the DNA is in the nucleus, it's called chromatin. Chromatin. This one comes again from the Greek chromo, which is color. And these are little colored bodies. They're not really colored bodies when we had a primitive microscope going back more than 100 years ago, close to 200. The primitive microscopes could only work with light, reflection and refraction on the light of the microscope. And so they created like little shades. The people who were seeing under the microscope, the very first nuclei, they noticed that they were little shady, they were off color because most of the cell is transparent, right? And the organelles inside the cell, they're transparent or translucent. But the diffraction created a little bit of a shadow, and that's why it was called the chromo. So the chromatin is the name for the DNA inside the nucleus as it is packaged with nucleosomes. It turns out that this is also known as beads on a string, kind of like a pearl collar. You consider a pearl collar, beads on a string, all right? The nucleosomes, the beads on a string, again, wind up again on top of each other one more time. So now we're into supercoiling. It's like taking a rubber band. You know, you always carry a rubber band in your back pocket because you never know when you're going to use it. And you take a rubber band and you twist it. Okay, so first level of twist, that's the double helix there. But then you keep twisting and just imagine that there's a nucleosome in the middle there. It's going to coil around that nucleosome, but you keep twisting and it's coiling upon itself and upon itself, some more bunched up, right? And eventually I can coil this into a little wad, all right? That's the supercoiling there, all right? And it keeps winding upon itself more and more and more and more like that, all right? And that's the supercoiling. Now, what's the point about this? The point about this is that this is called compaction, because the original rubber band that was this size, I have now bunched it up into a little tiny ball that is much smaller by size than the original stretched out rubber band. So this fits into a smaller place than the original rubber band. Okay, that's called compaction. What is the level of compaction of the DNA in the chromatin? We have to use a paper to illustrate that. So compaction or folding upon itself. If I fold the paper once, all right, so that's one fold. Let's say that's one level of coiling. That's one fold. The original plane was that plane, right? That's the original surface. By folding the paper in half, I have reduced the surface to half. But it's still the same amount of surface, but it's halved, right? So that's fold twice, fold again. How many pieces of paper do I have now? How many surfaces? 4, right? I folded twice. So this is a two fold, 3-fold, 4-fold, 5-fold, 6-fold. I can now go beyond 6-fold. You're welcome to try if you want to, but I cannot go into beyond 6-fold of a normal piece of paper, right? But you can see that I have reduced the size tremendously. Ignore the width of it for now. Just think of size, the folding from the original piece of paper that covered this much surface, okay? And that's only 6-fold. What is the compaction? So if we took out a DNA from a cell and stretched it out, and how long would it be? About 6 feet, right? Which this is 3 feet, so it'll be twice as long. One.
It wasn't meters.
It was 1 meter?
Was,
Well, a meter is 3 feet.
okay, I was going to say.
It's 2 meters, no?
3 meters.
Yeah, it's 2 meters, so it's 6 feet, or it's that long, all right? It's longer than this because 3 feet or 1 meter is 1 arm and 1 chest. It doesn't matter, it's about the same size on the other side too. Anyway, the point is that one single chromatin molecule, by the way, when the cell is in interphase, in other words, functioning normal, not reproducing, right? That splitting, the cell is interphase, is just one chromatin molecule, one DNA molecule inside the nucleus of each cell, one single with gazillion numbers of atoms that forms this. It's a macromolecule, it's a humongous molecule, but it's just one molecule. And that is called chromatin. In other words, it's not distinct chromosomes because the cell is not reproducing. Only when the cell reproduces does it make distinct chromosomes. When the cell is in interphase, in other words, functioning normally as a brain cell, as a liver cell, as a heart cell, it's just a chromatin molecule. If I take out one chromatin molecule from one nucleus of one of my cells and stretch it out, in other words, uncoil it, take out the supercoiling, it would be about 6 feet long, about 2 meters long. So what's the level of compaction? To take a string that is 6 feet long and to coil it and coil it and coil it and coil it so that it fits inside a nucleus of a cell. The level of compaction is approximately 10,000 fold, approximately 10,000, right? So you can see the difference between this. If we were to fold this paper 10,000 times, we could certainly fit it inside the nucleus of the cell, but we can't fold it that much. All right, so that's the compaction. So the point about all this is that DNA is super compacted, is basically shut down. Most of our DNA in most of our cells is shut down most of the time, not readable. And that's a good thing. That's a good thing because you think about it. What am I saying? Most of our DNA is shut down in most of our cells most of the time. Why? Because in every single chromatin of my body, I have the full genome, the genotype. I have all of the genes, the 10 to 20,000 genes that make up the human genome, right? The coding region, in every single cell of my body. And if every one of those genes expressed, I would be having too much expression. I would be having liver enzymes coming out of my heart, and I would have heart enzymes coming out of my brain, and everything would be expressing all over the place. It would be absolutely chaotic. We say it's non-viable. It's not sustainable. So it better be that most of the genes are shut down most of the time in both cells. And in fact, the vast majority are. And what happens? Only a little region of the chromatin is going to be expressed. For example, the genes coding for digestive enzymes is what's going to be expressed in the cells lining up the stomach. And the genes coding for neurons are the ones that are going to express in the brain, and so on and so forth. You can see the argument, right? So it's an important thing. This compaction is a very important thing to keep the cells really functioning properly, right? With cellular specialization. Okay, this is just kind of basic stuff about genetics and DNA, but it's all about the genetic sequence. This is what has the genetic code, this thing here, that codes for the 20 amino acids that make up all of the proteins in our body. 1125. Oh. Okay, so the genotype codes for the phenotype. The phenotype is the expression. For example, we either have attached earlobes or we have free earlobes. There doesn't seem to be any evolutionary advantage to that, unless you're a female, you want to, it depends where you want to dangle the earring, okay? But it's just aesthetic. It's one of those Mendelian things that our earlobe is either attached or detached. And there seems to be not much evolutionary advantage to it. But that's the phenotype, that's the expression. The genome itself, okay, so here is what is known as a karyotype. A karyotype is just mapping out the 23 chromosomes. In the case of the human, for any species, it will be their number of chromosomes. Karyotype. Again, the Latin type, or to show, and karyo, what's in the nucleus. Basically, karyotype is to match up the number of chromosomes. So is this karyotype from a male or a female? Or a man or a woman? What do we look at? We look at the sex chromosome, which is one of the pairs, right? 23 pairs. It's a male because XY, all right? Of the 23 pairs, one pair is the sex chromosomes, which is the 23rd pair. And the other 22 pairs have nothing to do with the sex, with the genetic sex. And so they're called autosomes. Autosome has nothing to do with the automobile. They're just autonomous from sex, basically. Okay, now notice the karyotyping, how it's done. You notice that each one of these pairs is like framed a little square around it. See it in the photo? Oh, yeah, very well. What is that square around each one of the pairs? That is the scissor cut, because this is actually a photograph of a cell undergoing mitosis. And there's a photograph of it, and that photograph is printed on a piece of paper, and then a technician will go and cut out, because the chromosomes are all over the place, right? And the technician will cut out each individual. It's actually individual. You see how there's a line in between the two here? Okay, very, very visible in chromosome in pair 1. You see the line? So they're actually individually cut out from the paper and then paired up with another blank piece of paper to make the match. And they're looking for gross abnormalities. For example, an excess number 21 chromosome. If there were a third one here, what is that? Down syndrome, trisomy, because it was discovered by this Dr. Down, okay? Or this is Down syndrome, which is a third pair, a third chromosome, 21, which happens to be the smallest chromosome. When they were numbered, the microscope was so primitive that 22 looked actually smaller than 21. And that's why it was named 22, it was number 22, but you can see here clearly, fairly clearly, that 21 is actually smaller than 22. So they should be flipped, but it's historical now, that's the way it is, and 21 is the smallest chromosome of the human genome. Not only that, it has the smallest load of genetic load of exons, right, of genes, which doesn't have to be, it happens to be the smallest and the smallest amount of genes in it. And so generally a third chromosome 21 is compatible with life. This happens to older women when they're their eggs are very old, after 32 to 35 years of age, they tend to stick during meiosis. And so what happens is that this aneuploidy is called aneuploidy, can stick any one of these chromosomes, but for the rest of the chromosomes, because they have more genetic load in them, that's not compatible with life. And what happens is that it is lost early on. It's an early loss in the pregnancy, typically a spontaneous abortion or miscarriage. And many times a woman doesn't really know because it happens in the first couple of weeks of fertilization, and they're not compatible with life. But trisomy 21 is compatible with life generally. They make it through the embryonic development all the way up to birth. They have a shorter lifespan and so forth, but they are compatible with life, with the range of abnormalities that Down syndrome people have, which are physiological and or psychological, right? And that's a range there also on both levels.
So the 13 and 18, which are the other ones they're commonly tested for, are also compatible with life?
Generally, they survive. They tend to survive less than 21. So proportionally, in part, but it also has to do with the genetic load itself. You know, the genes that are carrying, I don't know that much about it really, but they are somewhat compatible. What do you notice about 13 in general? That the P or the Q arms, the P arms are smaller, much smaller than the Q arms. I'm just seeing that right now. So something that strikes me. Compare, for example, 3, where the P and the Q arms are about the same size, right? So that may have something to do with it. I don't know. but that's one. And the same argument for 18, the discrepancy in the length of arms, which, all things being equal, very little load on this P arms here, right? And same here. Anyway, so that's a karyotype. Yeah, these are karyotypes. So this is basically just a demonstration for the genome, what makes the genome, all of these together. Next is all or none characteristics, which are really rare in nature, doesn't occur that much. Mendelian, when we do Mendelian genetics, it's basically all or none characteristics. But because it's a simplified model that actually occurs in nature, then it gave Mendel the possibility to propose the inheritance factors, which later was confirmed experimentally as being the actual genes in the chromosomes in the nucleus of the cells. Okay, so Mendelian genetics are all or none, meaning that they are not polymorphic, non-polymorphic. There's no range. The switch is either on or off. All right, some Mendelian traits in human, which are rare, and most of them are innocuous, or there may be some diseases. For example, the ones that are innocuous, that don't have any survival advantage that we know of, is where we have the hair in between the digits of the finger, right? Some people have hair in between the middle digit of the fingers, and some other people don't have hair there. That's all or none. Then another one is some people can curl their tongue, other people cannot curl their tongue. All right, that's all or none. So you can figure it out. You can do that. The widow's peak, which some people, and it's not just women, but it's this thing coming down on the forehead of hair. And some people have straight, other people shave it off, but they do have a widow's peak. And so that's another all or none characteristic. The earlobe we talked about. Oh, yes, twisting that thumb backwards, like some people do, like the trigger thumb. Most people cannot do that, but some can do that. And those are all or none characteristics. The Rh factor, the plus minus Rh positive, Rh negative in the blood is also all or none. Rh comes from the rhesus monkey. Rhesus monkey is an Asiatic monkey that is fairly tameable. In fact, they're becoming pests. They grow, they take over cities, and they herd. It's scary to see these hordes of monkeys coming into a town and terrorizing the people. But anyway, the rhesus monkey has been used for research, clinical research, because it has a fairly similar blood chemistry than the human, and that's where we get Rh. See, Rh factor is the first two words of the genus, rhesus. So now you know, and we are either Rh positive or Rh negative. What is it? It's a protein or a factor on the surface of the RBC, right? So if we have the protein, like the rhesus monkey has it, then we're positive, Rh positive. If we don't have that protein on the surface of the RBC, then we are Rh negative. Has nothing to do with the ABO factor, with the blood type. All right, blood type is different. That's another thing. Those are a combination of two proteins, A protein or B protein. Actually, I think it's a carbohydrate. It's also on the surface, but that's called the ABO group. That's for the blood type. And so there are two different types of carbohydrates on the surface of the red blood cell. They're called A and B. So if we have that carbohydrate on the surface, then we're A type. If instead we have the B carbohydrate, then we're B type, right? If we have both, then we're AB. And if we have none, then we're zero. Actually, with the O, it's actually zero. We have none.
And then within each one of those, you can have that.
Yes. Exactly. So, for example, I'm O negative, right? Most people are O negative or O positive, which you know our blood type and the Rh factor.
So that's about, so the negative and the positive in your blood type indicates the Rh factor.
Exactly. Which is a protein independent of the carbohydrates A and B. Yes, but they can also coagulate or not coagulate, because if I'm Rh negative and I receive positive blood, my immune system is going to identify that protein as a foreign object, and it's going to attack it, and it's going to coagulate it, and I'm going to form blood clots, which is not a good thing for health. And the same the other way. So a positive person can receive negative blood, but a negative person cannot receive positive blood for the Rh factor. And the same argument goes with the blood type, only it's a little more complex, because, for example, A people can receive A, right? And can receive O, because O has none of the carbohydrates. So an A person can receive A or O. That's why O is called the universal donor. B, same argument. B can receive from A, from B, and from O. AB can only receive from AB. Because if we receive, and no, sorry, AB can receive from AB, from A, and from B, and from O. So AB is the universal recipient. Yeah. And then the ones that are stuck are the O, because O can only receive from O. If I get an A-type blood, I will coagulate the A.
Is it the minority then? What would be the minority?
So O is actually the majority in the population.
Oh,
So
okay.
to compensate, yes, most people are O negative. Most people are O negative. Some people are O positive. You know, O positive is actually the best of the best, because you can receive from everybody. You can receive from any 4 blood types, and you can receive positive or negative, right? Because you're positive. So that's how the blood types work and the Rh factors. But those are all are non-characteristics. Either we have the Rh factor or we don't, or we have the A factor or the B factor or none, or both. Okay. Polymorphism, also known as quantitative traits, because they are on a range. And there it is. The faces of the cats can be innumerable. The heights and the color of the skin of people can be innumerable shades and tones, right? From the palest to the darkest. And those are all ranges, and they're known as quantitative traits. These are not palm trees. These are actually ferns. They're called giant ferns from Australia. They're kind of living fossils. They've been around for millions of years. And you can see definitely a range in size there. Guppies, these are all male guppies of the same species, and look at the variation on the fancy tail. Mushrooms, you name it. These are all, by the way, these are all the, what is that edible mushroom? The boletus. Yeah, these are all boletus mushrooms. And there's a wide range here, from the most edible delicacy to the most poisonous one. So nature, they're all quantitative, not all, but most are quantitative traits. And that's the distribution. So 5 theories of Darwin's evolution, or 5 points that led to the theory of evolution. It's 11:40. How about we take a little break here? We still have about 5 slides here, and then the second half is a little shorter. So this is a good place to stop for a moment. I'm going to leave it here, just mentioning them. The non-constancy of species. This was 5 observations that Darwin made that led him to the theory of evolution. Okay, the bottom line. This is the distilled. This is the high quality gin, fully distilled. 5 observations that he made over a period of years that led him to this theory of evolution, which is still the operating theory to explain the origin of species, of all species on Earth that we have discovered so far. First, non-constancy of species. Second, descent of all organisms from common ancestors. Third, gradualness of evolution. Fourth, multiplication of species. And finally, natural selection. I'm gonna break there for a moment, just 10 minutes. It's 11:41, so I'm gonna restart at 11:50. Okay, 11:50 gives us 9 minutes. So what I need to do, anyone remember? Pause the recording, Professor. Okay, resume recording. So what we're gonna do now is we're gonna walk through these. And these are the 5 observations that over some time Darwin made in his voyages mostly. And then thinking about them, how is it possible for one species to be able to generate another species? Again, the big mechanism here is natural selection. In other words, that somehow nature favors the individuals that are most fit to survive. So I have a slide for each one of these. The non-constancy of species. So I take each one. Let's see if this is going to work. Yeah, okay. So I have them. I have a slide for each one of these 5 observations, right? Non-constancy of species. If we look at the fossil record, which I mentioned already in previous classes, one of the main arguments in favor of evolution is the fossil record, right? We see bones, we see structures, we see imprints or the amber preserved little animals that got caught in there. And we see that they are similar, but different from the current species. And so there's some kind of similarity. For example, the elephant, which is current living, right? And no living ancestors. Keep in mind, if they're ancestors, they're from before by definition. In other words, our great grandparents, our great grandparents, they're not around any longer. They're ancestors. So the mammoth, for example, of which they're not just fossil, but actual tissue from the mammoth, preserved mostly up in Siberia in the cold, ice, and so forth. Similar, but not the same. The ears are much smaller than the current elephant. The tusk is much larger and curlier than the current elephant, and so forth. Those are just simple characteristics from the actual preserved animal, mammoth, embedded in ice up in Siberia. Then we go back further years. Trilophodon, for example, Trilophodon here. Here is an actual skull of a Trilophodon in a museum. There it is. You see, now it turns out the tusks are facing down instead of facing up. And that's an actual fossil. And here is a reconstruction of it about 25 million years ago. MYA, million years ago. And there's a fossil that is further back that is known as the Moeritherium. Okay, so that's the design, that's the drawing of the Moeritherium. And this is an actual fossil of it. There you have it. That's 35 million years ago. The tusks were not around. So these are the ancestors to the elephants. Similar, but different. All right. Non-constancy of species. When we go back, we don't find the same species. We find similar species. We go back in time, but they're not exactly the same species. Then, the second point, descendant of organisms from common ancestors. What is known as branching evolution, or the tree of life, or the phylogenetics that we can do today, precisely with percentage homology, right? Percentage homology, we can determine the 3 great domains of the bacteria, the archaea, and the eukarya. In fact, this is a more sophisticated classification done by phylogenetics. Look at where animals, plants, and fungi are at a very recent branch over here. All these other organisms, these groups, are similar but not the same. All these others, of which the eukaryotes are only these here. The archaea here on the eukaryote branch, and the bacteria are on this side here. So what is assumed from this branching evolution, if we go backwards in time, just like we turn the clock back on the universe, on the current universe, that it's expanding, then if we turn the clock backwards, then it should be contracting, right? To a single point, the point of singularity, which mathematically is one dimension, and in physics is singularity. All right, so the same we can do with the phylogenetic tree, go to the root, go to the trunk and the root, and then eventually we should be able to come up with a common ancestor, which is the oldest common ancestor of all life, from which life originated. Some kind of a protocell, like the fellow in the TED Talk was showing last lecture, of chemicals coming together and beginning to show the two basic characteristics of life. And the two basic characteristics of life are metabolism and reproduction. In other words, generate another one like themselves, right? And metabolism, which is basically chemical reactions in an organized fashion. That's the most simple expression. Going forward from there, the third characteristic that he observed, the gradualness of evolution. It's a process. This one has been studied extensively because a fair abundance of fossil record. Remember, there's a paucity of fossil record in general. Paucity is another one of those sophisticated words. It just means poverty in a more sophisticated way. Keep in mind, scientists, we invent all these words because we don't want people to understand what we're saying. Paucity, it just means there's lack of abundance, very few bones. And not only that, think about this. You know, we find a bone, we find a skeleton, we find a skull. Who's to say that that bone is the norm, is representative? Maybe it's the abnormal one, okay? We tend to use it as the norm. So El Hippo, right, which was the first considered dog, the precursor, not the first horse. I need to correct myself. The precursor species of the current horse, Equus, was called El Hippos. Goes back 50 million years ago. It was actually the size of a dog. It looked more like a dog than a horse. But it's an ungulate. In other words, it was starting to walk on its fingernails or its toenails, right? That's the front foot. You can see it has 4 toes. And then little by little, revolution, so through this natural selection process, the middle finger tended to survive or tended to get most of the weight of the animal. Maybe for running faster in a savanna area, I don't know. You have to think of the proper conditions that favors the middle finger overdeveloping and the other little fingers or toes being absorbed or becoming degenerate. If they're no longer touching, they don't have much of a function. And this is also a quadruped. Quadruped meaning it has 4 legs, actually needs to walk on all 4. So the 4 limbs, the forward limbs, the forward arms are not really arms, are not available for doing something else, right? So the fingers as such, as articulation, are not that useful when one is actually running through the savannah. What is more useful is a minimal point of contact for friction and for getting a good grip on that soil, the grassy soil. So apparently the best grip to run away from the tiger that's right behind me is the toenail, the actual nail, which is the hoof. Today, it's actually the nail. Imagine, compare a horse hoof with a human nail. Nothing to do with each other, but they are homologous. They come from the same origin, okay? And that's what the horse today stands on 4 nails. which is the middle finger of each one of the four legs. And the other fingers have become so degenerate that they're actually disappeared or reabsorbed into the body. And that's in 50 million years. So there must have been some kind of selective pressure on this animal to run faster and faster, whereby having four fingers on the ground for each leg actually made it harder to run, not as fast. Just speculating a little bit, because this is a herbivore, and therefore there's going to be an omnivore or a carnivore right behind it, right? Hunting it down. So gradualness of evolution is a stepwise process. For example, here, the emphasis is on the size of the animal and also the corresponding skeletal structures that go with that size. The fourth one is the multiplication of species. In other words, what we call today biodiversity. Look at the variety of butterflies. There must have been a single primitive butterfly from our ancestor butterfly, from all these butterflies developed or branched out. So branching evolution, right? Look at even the dinosaurs, which are now extinct. Actually, some people are making the claim that more than lizards, birds are the descendants of the dinosaurs. So basically, they're saying, no, the dinosaurs are not extinct at all. They just took off into the air and they're flying around. Okay. It's a theory. Anyway, the branching of dinosaurs was pretty evident from the fossil record, because each one of these creatures here is not invented by the human mind. There's an actual skeleton or fragments of a skeleton that correspond to each one of these figures. And you can see a branching effect over a span of about 100 million years here. So the drastic extinction, you notice the little triangles and diamonds and circles here. This is the end. This is the end line, right? Which occurred about 65 million years ago. And you notice it's a drastic, precipitous end. All of a sudden, all this variety of dinosaurs ended drastically. And there's a fossil record there to prove it, about 65 million years ago. It points to some kind of catastrophic event that occurred worldwide and wiped them out. And typically, the prevailing theory is that meteorite that hit Earth around what is today the Yucatan Peninsula and is buried down there about 5 miles into the crust of the Earth. The fragments of the meteorite are still there. They created such a huge amount of dust and fire throughout the world that it wiped out the dinosaurs, okay? Probably wiped out the herbivores first, and then the carnivores that were feeding on the herbivores. But you can see here, it was drastic, catastrophic end of this biodiversity of the dinosaurs, but they can be traced back, again, phylogenetically, because of the fossil record, to fewer and fewer ancestors in the branching effect, if we go back down into the trunk, to just 1 or 2 individual skeletons or individual species that gave rise to all these branches. In fact, you see 3 that look very similar. Let me see if I can do this. 3 of these guys that look very similar, but gave rise to 3 separate huge branches. Look at these 3 guys over here. The Herrerasaurus, the Saurodomorpha, and this one is unnamed, but see how similar they look? They're all bipedal, right? In other words, they're already running around on their hind legs, and the front legs are free and starting to become degenerate, kind of small. But 3 main branches, one down here, the one in the middle, and the third one up here. All these look similar to each other, long tail to compensate for the pivoting on the hind leg, on the pelvis, right? And from all this, the variety, this may go back about 200 million years ago, more or less. So this span took place in about 150 million years. to the three branching varieties. The one at the bottom here, see? Some of them, their forward legs regenerated, they went back into forward limbs to walk on all fours, the Triceratops and all those guys. Others remained, I guess this branch all eventually went down to 4 legs. The middle branch also, this is the Brontosaurus thing that is herbivore, probably the largest mammal on Earth, on land that ever existed. Whereas the third branch up here, they're all 2-legged, and most of them are fast running with huge jaws, which means carnivores, not herbivores. So you see the variety and the balance between them. So these guys were feeding on each other, and the herbivores were, of course, feeding on the plants that were around. The point here is the branching effect. So multiplication of species with time, and not vice versa. There's extinction, yes, as a dead end, but we don't see species merging. Rather, we see divergence. A little bit similar to language. We can see that newer language developed from previous languages, the 5 Romance languages that come from Latin, and Latin coming from Greek, and so forth. That's a behavioral thing, right? That's a cultural thing. All right, the fifth one. Oh no, here's another example. Okay, this is one on the mammals, the same branching effect, not just for those people who say, well, that's historical, that stopped 65 million years ago. Well, here's one that's current on the mammals. Or the branch, I think there are 28 or so branches, families of mammals today, right? They go back precisely 65. Look at the coincidence. It's not coincidence. Is that when the dinos wiped out, which were incredible carnivores, ate everything in sight, at that time, the only mammal that was around was really a little tiny thing like this that scurried around the grasses and the bushes and mostly hid under rocks and caves. It was like a little hot dog with 4 legs. That was the primitive mammal with fossil records of it. When the dinosaurs disappeared, then there's a branching evolution of the mammals. There's a flourishing because now those little hot dogs came out and say, oh, no dinos today, finally, we're free. And so they started branching out. And we get all the species of mammals from the single common ancestor, of which there's a fossil record back then. And it coincides, it matches up with the disappearance. So we have these mass extinctions followed by expansive radiations. All right, expansive radiation, which is this effect right there on the mammals, with all kinds of fossil documentation for it. Next. All right, that's about it, because the fifth one is actually natural selection, right? And natural selection is what we're going to get into in the second set of slides here. Yep. So here we go. Some of it is going to be a little repetitive, but here we're going to look at the 4 main forces that may cause evolution. Again, we're going back into a probabilistic model. Now, when you read Meyer, he mentions 7, but 2 or 3 of those can be clustered. Like, for example, he has mutations separate from recombination and tandem effects on the DNA and so forth, that I clustered them all together with the genetics of mutations in general in the DNA, all right? So mutation, migration, drift, and selection are the 4 main forces that may cause evolution. But the one that actually does it is selection itself. The other 3 predispose the organism to may be selected or not. The other 3, in other words, we may have mutations and not be selected out. By the way, selection is a selecting out of the weakest, right? So we may have mutations and not be selected out, or we may migrate and not be selected out, and so forth. So let's get into each one of them little by little. First, the review of what I just talked about. You see, he lists this one individually. That's how he comes up with 7 forces. but I distilled it down to 4. I'm clustering mutations, recombination, and transposons all as one cluster, generally under mutation, because one way or another, they cause a change in the DNA. They cause a change in the DNA. Now, it occurs mostly in sexual reproduction, but it can also occur in asexual reproduction with something that is called lateral gene transfer. This is for bacteria as they share genetic material between one another, bacteria of the same species, obviously, or the same type. And then clones, also in cloning, is asexual reproduction, of course, because there are no gametes involved. But there can be mutation occurring in cloning, which could cause speciation, can cause evolution. Now, in sexual reproduction is the most prevalent one. It happens with the gametes at the level of the cells reproducing, undergoing meiosis. And meiosis is like a double mitosis, right? So let's look at meiosis for a moment. First, the reproductive organs, in this case of the human, that are very important organs, but they're not vital organs. Okay, they're important for the next generation, but they don't. They're not vital for the individual carrier of the organs. So we know the ovaries making eggs or ova, and the testes making sperm. This is an actual micrograph. It's a microscopic photograph. You notice that the sperm is a single cell, of which each ejaculate has millions of them, actually hundreds of millions. But the egg is not just a single cell. The egg is actually a cluster, it's a cumulus, right? That has 3 layers. It has the actual ovum, and then it's surrounded by a zona pellucida, and then a corona radiata. So it's actually a cumulus of cells that is protecting the egg as such. Where they're made, so the eggs are made in the ovaries, specifically the follicles that mature on the cycle, and then the follicle ruptures, and that's the actual ovulation process. Here's one in the process of rupturing, and then the follicle that is left inside becomes a corpus luteum, which also provides hormones for sustaining the pregnancy if there is a pregnancy. Whereas in the sperm is the seminiferous tubules that produce the actual sperm that are stored in the vas deferens, and the epididymis. These are all the little network of tubules that are inside the testes and through the penis, where millions of sperm are accumulated for ejaculation. Now, you notice that whereas a woman is born with a set number of eggs, the baby girl has a set number of eggs, which is in the millions in each ovary at birth. They are in arrested meiosis. I'm going to cover this in greater detail next course on the beginning of human life. But those eggs are arrested, and then in a dozen years, more or less, they start maturing with menarche, with the first period, and continue throughout the reproductive years of the woman until menopause, when she runs out of eggs. In contrast, the man is begins producing sperm from scratch, from precursor cells inside the seminiferous tubules. Again, during the teen years, usually 1 or 2 years after the woman, more or less in the early teenage years, and will continue to produce sperm for the rest of his life until death. All right. So why don't we just explode with XX sperm? Is because sperm normally lasts only 24 hours. That's the normal lifespan. Just like the egg, also a mature egg only lasts an average of 24 hours. And then they become overripened and they start degenerating. They're digested away and reabsorbed by the tissues, by the tubules themselves, reabsorbed, and then more sperm is produced. So basically every 24 hours, sperm is new sperm that is produced all the time for the rest of the life of that individual. Okay, so those are basic differences just in the production of the gametes. The important thing there is that they are haploid, so that the complement needs to happen at fertilization. Asexual reproduction, which we're kind of less familiar with, happens in a number of organisms, plants and animals. Bacteria also. Bacteria have little tubes that are called pili, one pilus, several pili. Those pili connect one bacterium to another, and then a transfer of DNA, which typically is a circular DNA or a plasmid, it's called plasmid, right? Can be transferred from one DNA, from one bacterium to another. So it's a type of a lateral transfer, lateral gene transfer, or in plants and animals, cloning, which in plants is called vegetative propagation. The lawn, on your backyard, if you pull out one tuft of grass and you get a whole string of them like that, right? Those are all clones. So the little string, if you will, is a little shoot that comes out laterally, goes into the ground, and will put out roots, and a new plant will come out of that. So this is asexual reproduction, non-sexual reproduction. There's no mixture of gametes, and therefore they're all clones of each other. They're almost identical to each other. This is the argument of the identical twin, right? Happens in plants by vegetative propagation. It happens in animals by budding, for example, hydras and jellyfish and things like that. It happens with amoeba in pinching in the middle, bilateral fission or binary fission. It happens in starfish, for example, fragmentation and regeneration. When a starfish loses an arm, it will regenerate that arm. Generally, it will not grow as long as the other arms, kind of like the tail of a lizard when the tail breaks off. But also the arm can regenerate a new starfish, as long as the arm contains part of what is known as a central disc. The central disc is what goes around the middle, what connects all the arms together. Because that's where, what do you think is there at the central disc? The stem cells, the adult stem cells that regenerate either the arm, the new arm that broke off, or the other arms from the arm that broke off. All right, it's called fragmentation and regeneration. They're all different examples of cloning. Asexual reproduction, where you get from a single individual, you get more individuals that look very similar, but they're not identical. Okay, other types of mutation, they can be duplications, deletions, additions, moving around, jumping genes, all right, transposons, they're known as transposons. So all kinds of different types of mutations that would occur naturally, normally. Most of these fall on the 97% or 98% of the junk DNA, so they're neutral, they don't have an effect. Okay. All right, so I have a few more here. It's getting a little too detailed. Recombination also, it's a crossing over event that happens during meiosis for the production of gametes. Again, too much detail, I'm not going to worry about it because I'm going to cover this in more detail when we get to the beginning of life course in the summer. But just know that these are, we can put them within the category generally of mutations at the level of the DNA, change in the DNA, so that the gametes can produce mutants, right? Or recombinant. See, these would be recombinant gametes here with the blue and red, whereas all blue are all red. They're non-recombinant, and these are recombinant chromosomes. All right, transposons, there's a little film there that you can see about it. I'm not gonna worry about it. It's too much detail. Even viral infections can also be considered a transfer because the virus injects either a DNA or an RNA, which then sequesters the mechanism, the replication mechanism of the host cell to replicate itself. And it goes through the lytic and lysogenic cycles of the virus back and forth. That's why a virus technically is not considered alive, living, because it cannot reproduce by itself. And it doesn't metabolize either. So really, as far as nature is concerned, a virus is closer to a crystal than it is to a living organism. Because it's just very highly structured molecules, huge organic molecules that make up the virus, but by itself is neither metabolizing nor reproducing. Necessarily needs to infect, parasitize a host cell to make more of itself. That's the virus. Okay, anyway, any mutation, any change in the DNA sequence can be put under that thing of mutation. A lot of these mechanisms occur at random, all right? So there's no real predisposition to it or trigger. It can just happen. There are insults, or called insults, that can cause mutations in nature, and those insults can either be chemical or physical, mechanical, or a combination. For example, UV, ultraviolet radiation, especially UVC on the skin, can cause skin cancer, which is a mutation of the DNA, right? On the epidermis or dermis. Oops, wrong slide. And that's an example of a chemical insult. It could be heat also, it can cause mutations. All right, second force, gene flow or migration. Gene flow or migration. There are different types of migration, two types, immigration or emigration, right? So if we're going into a territory, it's immigration. If we're going out of a territory, it's emigration. And this applies for all other species, animals, plants, fungi, and bacteria. So it applies to all the major groups of living things. In the plants, for example, I cannot speak because it's a PDF. Sorry. Okay, but in plants, oh, it's another way I can make it big. Hold on. I can do this. Yes. There's a will, there's a way. I'm going to lose resolution, but it can still be seen. Let me cut it down a little bit. Okay. Now the question, oops. The question here is, how many species of plants are there on this dune? Now we can tell at least 2, right? There's this creeping thing over here on either side of the dune. And then there are these tufts of grass sticking out in the middle of the sand. So we can say just at face value, at least 2 species are on this dune. And this is what are known as pioneers. They come into a barren area like sand, all right, and they establish themselves. What's your intuition about the root system of these guys? Rather extensive down there, especially going down where they can find some humidity to survive, because the root, the water has to come in through the root system, liquid water, but even a little drop of water will do, a little humidity around some sand grains will do. So the intuition is that these plants have roots, very fuzzy roots that go fairly deep fast, get established, because at the surface it's going to be dry, but deeper down, there'll be some humidity accumulated down there, right? Okay. So we see 2 species. They're immigrating. So these are pioneer species. These are immigrating. Remember environmental bioethics, the pioneer species. They are immigrating into a barren area, otherwise known as sand, a dune, right? So that's an example of immigration. The other one is an example of emigration, is zebras moving out. But I don't know if I can move this sideways. No, I can move it sideways. Use the arrow. Okay, so here is a herd of zebras. If I push the arrow to the left, they move to the right, or vice versa. Yes, this is a herd of zebras, can be seen, right? Running from right to left, all right? And you say, oh yeah, okay, all these zebras are alike. Yes. But they're not clones, first of all. They don't have their parents, sexual reproduction. And secondly, they are similar but not identical by any stretch of the imagination. Look at the pattern of the stripes, right? Oops. Yes. The pattern of the stripes. I have to do this slowly because otherwise we'll go into the next slide like that. Therefore, I need to tone it down. Okay. Well, anyway, okay, so we have to deal with this. Look at the pattern of stripes here. So different from these two, right? Here above the belly, in the mid-region on the side. And then look at this pattern. Different again, they have one in the middle. And this other pattern is different, and this one is different again. So, you know, it's similar to this one, but not the same. So it turns out that every zebra has actually a slightly different pattern. There may be some selective advantage or disadvantage to that, right? But when these zebras move from one location to another, they may go through a bottleneck event where only some zebras got through and other zebras did not get through. And so drift, or sorry, gene flow, you can think of species, when we talk about migration or gene flow, think of species, individuals as gene bags. They're gene bags that are moving from one place to another, okay? That's a way of thinking about it when we talk about migration. Range of migration. Here is the sloth. So sloth is also a vice, which is moving slowly, or vagancia, not wanting to do anything, sitting in front of the TV, so known as a couch potato, right? Okay, so there are two-toed sloths and three-toed sloths for obvious reason. Which one is this? A three-toed sloth. Okay, I have actually seen them in Panama. Once we were on the road going through the countryside, and there was one trying to go across the road. It took the thing about 2 hours to go across the road, just 2 hours. The whole traffic was stopped because the giants, the sloth, not giants, just normal sloth, they're very awkward. They seldom move more than maybe 100 feet from the place of birth when they were born, and they just hang around those trees for the whole lifetime. Yeah, move very little away from they were actually born. So here's mother and kid getting a ride. That's why he's so happy, because he's getting a ride from mom, who is doing all the heavy lifting. Anyway, that's the sloth, very narrow range, and it's a mammal that is fully mobile, but very narrow range. In contrast, here's a pine forest, and it looks mostly the same species of pine, which has a huge range, and they don't move at all. But their seeds move, because the seeds are winged seeds that can fly all over the place. And so the contrast, a mammal who certainly has 4 legs that can move around has a very narrow range of population, whereas a tree, a pine tree that is anchored on the ground has actually a huge range of population, because the seeds are wing dispersal. All right, so that's with regards to range, migration, range. It has to do in part with behavior, the 4 different things that we talked about earlier. All right, it's also a dynamic state, especially on the borders. Here is a patch of, I think they're just maybe daisies, yellow daisies, but you notice over here there are violets, and in between there's also this white fuzz here. So I would say from just the flowers, there are at least 3 different species of plants here. The yellow daisies, the violet violets, and then there's white fuzzies in between. And there's an interface, right? There's a larger cluster of yellow back here in the background. There's a larger cluster of purple in the foreground, and the white ones are kind of in between, on the boundary between the two. So it's dynamic. The borders tend to be more impacted by diversity on the borders. Same with bacteria, for example. Here's bacteria in a dish, and they're showing what is resistance and susceptibility to penicillin, for example, which is produced by the other bacteria. But you notice the edge is whitish. So when I put antibiotic onto this culture, the ones that would be affected the most are the bacteria on the edge and not the bacteria in the middle. All right, so it depends also location. It's just like real estate. The 3 laws of real estate. location, 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 right? Where it's at. So it'll be a dynamic state depending where we're at the center of the cluster or at the edge. Which zebra is in more danger of getting eaten by the lion? The ones that are at the edge of the herd, as opposed to the ones in the middle of the herd. And in fact, many herding mammals do that. They put the calves, the young ones, in the middle, and the old bulls will go out on the outside, protecting the calves. It's just a herding behavior. All right, the same for plants, starting with the pioneer species that I mentioned on the edge. I think we covered this already in the environmental bioethics course, right? It moves from pioneer species through successions until we get finally to the climax community, where there is one or two main species of trees, which is going to be the largest, tallest, and most abundant tree, one or two species alone for the whole forest, right? That'll be the dominant species. Okay. The third force is drift, which is different from gene flow, because gene flow is migration, is that bag of genes moving from one place to another. Here with drift, what we throw into the equation is chance. In other words, where the individual that actually gets to mate is going to be. All right, so it's a chance event, and it's statistically is known as a stochastic process. Stochastic is a random chance event. For example, I mentioned the male gorilla that is about to mate, and a rotting tree fell on top of his head and knocked him out, and he did not get to mate. He happened to be at the wrong place at the wrong time. Okay, but it's a chance event. It's not that he planned, okay, I'm going to be under this tree now that it's about to fall. No, there was no clue. So basically, chance is inherent, and it's a philosophical thing. You think about it. Chance is integral. That was also one of Einstein's great contribution to his general theory of evolution, of relativity. That's why relativity, because chance is built into the system, into the planetary system, into the galactic system, into the universe. There's a chance, so it's a probabilistic model. That's why the only thing that really exists is mathematics. It's a very philosophical thing, but if you think about it, the chance of the individual being at the right place at the right time with the right temperature and environmental conditions, chance is built into the system, right? So there is randomness that is part of the equation of evolution, and that's known as genetic drift. Okay? This is to point out also populations. So for example, the lungfish here, these are called wrasses, and they're yellow wrasses, which can vary from bright yellow to bright orange, and anywhere in between. And then this other flat fish here is called a tang, and these are mostly blue tangs that you see here. But you notice also variation. Look at this blue tang with a dark face, and look at this blue tang with a light face. Who knows which one? Let's say that these are the male, because generally the female selects on the male. I don't know, the female may have a preference for light face as opposed to dark face, or vice versa. We don't know, but they know, okay? And so selection is going on here, depending on which fish happens to be in front of the female at that time. And it has to do with temperature and currents and feeding habits, and the complexity is just bewildering, but it happens. And chance is part of the equation of evolution. It's known as drift or genetic drift. Let's see. Okay, we only have a few more slides. It's 12:40, 5 more minutes. Okay, I want to talk about this bottleneck effect. And again, it's a probabilistic thing. If we have a bottle full of marbles with an even number of blue marbles and yellow marbles, and we pour those marbles into into a container, all right, into a container. By chance, the few marbles that happen to be at the neck of the bottle are the first ones to come out of the bottle, all right, and they're going to fall into the container, into the glass, into the cup, and they happen to be in uneven distribution because there were a few more blue marbles than yellow marbles at the neck area, just by chance. The distribution overall is 50-50 of yellow and blue. But at the neck, locally, where the bottleneck event happens, there happen to be a few more blue marbles than yellow. So what went into the glass was more blue than yellow. Now the ratio is, let's say, 4-to-1 or 3-to-1 or 75% blue and 25% yellow. When these individuals reproduce, they reproduce more of the same. So they skew the population now in favor of blue. And it's just because of the bottleneck effect. It's a random effect, all right, because all the marbles have the same size. They just have different color. And it happens that at the bottleneck event, at the bottleneck place, the local distribution there was skewed, and that's what perdured after the event. Again, this fellow was walking around, couldn't care less about beetles. He was not selecting out green beetles on purpose, all right? He couldn't care less about the beetles. He was just crunching beetles as he went along the way. It happened that the green beetles, which were fewer, got in the way of the shoe, so that skewed the population tremendously. This is an extreme bottleneck effect because if you notice, in stage 1, so the 3 stages of the beetles here, right? Stage 1 of the beetles, there's more brown than green to begin with. So fewer green. Then when the fellow walked around, it was at night, he wasn't paying attention, he was just crunching beetles, and he happened to crunch 2 out of the 3 beetles, the green ones. So he basically wiped out the population because he left a single green beetle and the brown beetles remain intact, so they continue to reproduce peachy keen and hunky-dory, no problem. But the green beetle alone, they happen to reproduce sexually, no mate. So it doesn't matter if he was male or female, they're done. So that bottleneck effect actually caused an extinction event. That was an extinction event for the green beetle. It was drastic, all right? It's not always that drastic, but bottleneck events have a strong effect, may have a strong effect on the resulting population played forward with time. And that's the drift. Okay. It can happen with biotic factors or with abiotic factors. Again, you're familiar with these terms, but just to recall them, biotic factors are biological factors. In other words, that the one that is causing the selection, that is causing the selecting or the drift or selecting pressure is actually a living organism. In this case, it's the cow feeding on the grass. This is very graphic. You see that on one side of the fence, there's plenty of grass. On the other side of the fence, there's very little grass, right? Because the cows are feeding on the grass, so they're constantly cropping the grass on the left, but because they cannot get to the grass on the right is very happy growing wild with no pressure on it, okay? So in this case, the drifting effect is being caused by a biotic factor, which is the cow on the grass. The cow, in other words, is the bottleneck, it's causing the bottleneck. But it can be a non-biotic factor, a factor from nature that is not living, like a lightning. It happens that this tree, which is, we can see from the photo, it was the largest tree in the area. Look at the size compared to a house, okay? By far the largest tree in the area, and it got taken out by lightning. So that tree is no more, will not reproduce going forward. So the strongest, largest tree actually got taken out by a chance event of a lightning striking right at that spot. So biotic or abiotic factors can cause a bottleneck effect or drift. at the microscopic level, it can happen at the molecular level, etc. So I'm just giving you examples of how it can happen at the various levels, going from the molecular, physiological, morphological, or behavioral. In the case of the cow, of course, it's a behavioral thing, right? I think just one or two more slides. Okay, so summary, finally. We have the four major forces of evolution that drive evolution: migration, mutation first, migration, drift, and finally natural selection, the fourth, which is going to be the next lecture. So I'm leaving that one, devote a whole lecture to selection and how that mechanism works. So this is just a summary, but you can see that chance is really built into it at all levels, and it depends on so it's a combination, just like it's either, you hear the argument of nature or nurture. Well, it's not either or, it's an and. It's nature and nurture, right? Nature meaning the genetics, the genotype, and nurture being the environmental factors together make the individual who survives or doesn't survive. Okay, folks, that's it. Again, thank you for your patience and perseverance. That's all I have. Questions, comments? Oh,
No questions here, Professor.
okay, thanks a lot. Thanks for being here. And now I'm gonna finish this and we will have our next lecture next Saturday. Just check that. Yeah, okay, so going forward for a minute on the schedule, let me just shut this down. I mean, start the download. So give me a minute while I close this.